Ja, sehr geehrte Damen und Herren, liebe Kollegen, es ist mir eine besondere Freude, Sie zu den Präzisions-Therapie-Symposium begrüßen zu dürfen. I will now switch to English because we have English speakers and it's a pleasure that we got the opportunity to invite outstanding scientists who are well aware of the progress of immunotherapy in rheumatic diseases. So I will chair this symposium. My name is Georg Schett. I am the chair of immunology and rheumatology at the FAU in Erlangen, and I will chair this meeting together with Gerhard Grönke, who just joined the symposium, and we will alternately introducing our guests. We have thought that this topic would be very important in the spectrum of immunotherapy because there has been a dramatic progress in the treatment of rheumatic diseases in the last 10 to 20 years, particularly based on the development of targeted therapies. And I would say in the last 10 years, there has been an enormous progress in better understanding the features of arthritis, but also other diseases like scleroderma, which is worth to discuss in this symposium. So we have selected 4 outstanding scientists covering different topics in rheumatic diseases, and we want to start with Constantino Pizzalis. Constantino Pizzalis is working in London, and he's obviously Italian, an expat, and he has done tremendous work in understanding synovial inflammation in rheumatoid arthritis, and has set up, together with colleagues in the UK, an outstanding collaboration to characterize rheumatic diseases on the tissue level. There has been efforts before, but it has never been done in such a structured and collaborative way like in the UK. And Constantino has actually set this up, and it's a pleasure, Constantino, to welcome you to this symposium. You term your lecture Portraits of Rheumatoid Arthritis, and I think it's a wonderful title, and I'm happy to have you here and look forward to listen to your lecture.
Thank you very much, Georg, for the very kind introduction, totally undeserved, but I take it. Thank you very much for inviting me to this wonderful symposium. Yesterday, I listened to outstanding lectures in the field of oncology. And one of the things that I would like to do today is really to, in a sense, show where we are in terms of developing similar approaches to inflammatory arthritis, particularly rheumatoid arthritis. So these are my disclosures. And I will start by asking the question, why do we need a new taxonomy of disease and precision medicine in rheumatoid arthritis? The reason for that is threefold. The first one is because the disease is highly heterogeneous, both from the clinical point of view, from the pathobiology point of view, which I will show it to you and discuss in more detail, and from the treatment response point of view. The second aspect is because approximately 40% of patients do not respond at all to current medications. And very importantly, we are unable to predict before we give a specific drug. And we are referring also to targeted therapies, biologic therapies, is that we cannot predict whether they are going to respond or not respond. The third reason is because we cannot afford to continue to use expensive medications such as biologics on a trial and error basis, because current approach really leads to poor response rates and decreased quality of life and increased toxicity and disability. And this is the cost of the disease in the UK alone. This costs £800 million of biologic therapies, but most importantly, the social economic cost in terms of sick leave and work-related disability is 4.2 billion. And because we cannot predict a current treatment response to any of the medications that we have available, this is the current paradigm, that we start with conventional synthetic DMARDs, and then we cycle the patients from drug to drug until we find the one that it works. Now, this time lapse until we find this drug that works is not inconsequential. At each step of the escalation from synthetic DMARDs to the first biologic, which is normally TNF, to other biologics, we have 40–50% of patients who do not respond and continue to have active disease. And by definition, then, if the disease is not controlled, the disability increases, the quality of life decreases. And so, although we don't see patients like this one any longer completely disabled, when we look at really our impact, which has been tremendous with the advent of biologics in patients who really respond well, then patients can go back to work. However, when we look at the proportion of patients across a spectrum of drugs, all those available are described here in these Nature reviews of Joseph Smolens and colleagues. When we look at the improvement in the disease response to the 70% mark, the ACR 70, as you can see that apart from few cases in early disease before the conventional synthetic DMARD approach, we reach 70% improvement in patient in 20–25%, whether we're using B-cell depleting agents, whether we are using co-stimulatory blockade agents, whether we're using anti-TNF or IL-6. So this is a real unmet need. So 75% of patients really do not improve to a level that is in our reach for some patients. So we need better biomarkers to define the disease, particularly in defining treatment efficacy and predicted biomarkers of response. Now, there's been a lot of work in the last 20 years in this area, and genetic work from Ann Barton and their group really is reported in this review. And you can see that a number of genes have been shown to be associated with treatment response, and this is go from methotrexate to etanercept and other medications. However, when we consider the real impact of the genetic contribution, this at best is 20%. So there are a number of other factors which we need to take into consideration if we want to understand why some patients don't respond to the first medication we give. And looking at the peripheral blood, this evaluates 57 eligible studies, 14 biomarkers were considered, and the conclusion of this meta-analysis is that no peripheral blood biomarkers are of sufficient predicted value to be of use in clinical practice. And so one will wonder, what is the reason for this? And I think one of the reasons is shown in this slide, which comes from Berlin, the Charité, and is almost 20 years old, in fact, 21 years old, and which shows that when you look at the transcriptomic profile in the synovial tissue versus the same gene sets in the peripheral blood, you can see that you got over 1,000 genes which are barely expressed in the peripheral blood, a very low level, and 200 genes also which are not expressed at all. So you have a signal in the tissue which is not being picked up in the blood. So several years ago, we put forward this hypothesis that perhaps if we were to look specifically in the synovial tissue, rheumatoid arthritis is a disease in which a proliferative synovitis really drives the joint damage, and work from Georg in particular has shown the relationship between the activation of osteoclast and the joint damage. So that this is the tissue which really mediates the diversity of clinical phenotypes and treatment response. And in fact, we look at the biopsy of approximately 100 patients with rheumatoid arthritis, all meeting the classification criteria for these conditions, clinically very similar, with a disease activity of an average of 5.8 DAS28, and so very active disease. And yet we could see three different subsets of patients in this group. First of all, a group that we call lymphoid pathotype, which affects approximately 40% of patients, in which you can see using conventional immunohistochemical markers, you recapitulate almost follicle-like structures that you see in secondary lymphoid organs. Now, this group of patients are very rich in B cells, but also all our immunophenotypes are strongly represented. Then there is another group of patients, again, significant numbers, 40% of patients, in which you can see you have a significant and major staining for the CD68, the macrophages in the diseased tissue. But when we look at the CD20, the B cell marker, really there aren't any B cells in these patients. Likewise, the third group of patients that we've identified, we call it fibroblastic or post-immune. There are very few immune cells infiltrating the synovial tissue, but these patients have high levels of inflammation just the same, and therefore we are postulating that the stromal reaction here is what causing the problem. Now, this is now the background for you to see where I'm going in terms of the treatment response. So why would you give rituximab to patients like these ones here? You wouldn't give rituximab to the non-B cell lymphomas. Likewise, to use another oncology analogy, you wouldn't give tamoxifen to estrogen receptor negative patients. So why we are using rituximab in a blind fashion in patients with rheumatoid arthritis? And at the end of the talk, I will present the data of a randomized clinical trial, which has just been accepted by the Lancet, in which we are really testing this hypothesis regarding the absence of B cells and the response to rituximab. At the time, we use microarrays, and you can see here that the histopathological grouping is also recapitulated in the molecular gene expression in these patients, in which we clearly have in this heat map diverse genes which are expressed and associated with the cellular infiltrate. The year after we published this paper, the group from Genentech in collaboration with David Fox in Michigan, they looked at 49 patients from joint replacement tissue, and then they reported similar results with the lymphoid and the myeloid pathotypes. And as you can see here, when they extracted cells and they do FACS analysis from these patients, you can divide almost the two groups of patients with rich in B cells or some B cells and patients with really poor B cells. So you can almost grossly divide patients into B cell driven synovitis and a non-B cell driven synovitis. And I'm sure that all the rheumatologists in the audience will share the concept that the CCP positive disease is a different disease from the CCP negative. So we have a similar distribution in the synovial joint, in which we have B cell rich patients and B cell poor patients. And again, looking at microarrays, you can see that there is again a stratification of these patients according to the gene expression. So we refined our hypothesis that we could really do molecular profile as well as histopathological profile in these patients, and therefore define potentially what are the pathways associated with the current targets of the biologic therapy. So if we had, for example, a patient with a prevalent molecular profile for IL-6 blockade, we could give the tocilizumab or other IL-6 blockers to these patients, and so on. So we also developed a technique which is a minimal invasive ultrasound-guided biopsy, which enables us to collect tissues from all patients and all joints, large and small. As you can see here from the histopathology of some of these patients and the agile and profile with the 2 bands of ribosomal RNA, which shows high quality RNA for molecular profiling in these patients. And we embarked then approximately 10 years ago to the program that Georg was referring to early, in which we classify patients and we enter them into trial synchronized for disease stage, early versus late disease, and for treatment response, or sorry, for treatment exposure. And this is very important because many of the published literature is not well synchronized with regard to treatment exposure. And we know that the disease pathology is responsive to treatment and can be modified by treatment. So the early arthritis patients were only DMARD naive when they were recruited to the trial. The established patients were DMARD inadequate responders and they were coming to the first biologic, whereas the third group were patients who had failed anti-TNF therapy, so the first biologic in these patients. All patients had a synovial biopsy prior to treatment and either were treated with conventional synthetic DMARDs or randomized to rituximab, tocilizumab, or anti-TNF in the STRAP trial, or rituximab and tocilizumab in the R4RA trial. And the R4RA trial, I will give you more details later. In all patients, we did digital pathology as well as molecular characterization. In the first instance, we moved towards RNA sequencing. And we constructed a very significant biomedical resource in which we have 768 patients that are recruited now to these trials, and with biopsy in all patients at baseline, and 42% of patients at the second biopsy. And important, we have in all these patients longitudinal blood sampling, as you see there. So the real purpose of this program is really to deconvolute the disease, and through a multi-omic approach comparing blood and tissues, we want to define what are the molecular mechanisms at each individual patient level in order to define disease endotypes and drug-related endotypes. So we want to build a molecular profile pre-treatment at baseline, which then hopefully will inform us on what are the molecular patterns in the non-responder patients, this red box here, versus the responder patients. And we know that approximately half of the patients respond and half of the patients do not respond. So they are very well balanced. The other question that this program will enable us to address is whether the patterns of non-response, for example, to rituximab are the same or different to tocilizumab or to anti-TNF. And the idea here is that if it's the same, in other words, we have the same pattern of non-response, the gene expression profile for non-responder patients, then probably we are dealing with a disease endotype. Vice versa, it is possible that the drugs themselves might influence the pathology, and therefore we want to identify whether there are specific pathways which are still amenable to the alternative inhibition with the alternative cognate drug. In other words, for example, if patients do not respond to rituximab, do we have pathways in that patient that will tell us that will respond to tocilizumab? And we can confirm this in the second biopsy of these patients. So the idea is that the second biopsy will tell us which pathway are still active in these patients. So let me tell you a little bit what we have done with this program now, and we're starting with the early arthritis program, which is called Pathobiology of Early Arthritis Cohort. And this study started in 2009 and completed in 2015. And we published the first 3 papers in 2019. So this is the first of these published in Cell reports, which confirms 2 things. In early arthritis, we still see, so not in established disease, not in late disease as the Genentech group published in joint replacement tissue, but in early disease at presentation, that we have these three groups of patients, the lymphoid, myeloid, and fibroid. And although these are not pure pathotypes, they are overlapping, and this is actually shown well in the RNA sequencing that you can see on the right here. Still, we have at the beginning of the disease, with the technologies that we have available now, confirmation that actually patients who meet the criteria for rheumatoid arthritis and have active disease, they show a different pathology and a different molecular profile. Miles Lewis in our group really led the bioinformatic efforts and developed a number of IT tools. One is shown here in this rotating volcanoplot, which you can interrogate on a database which is now being made available to all the community. And you can, for example, select your gene of interest. And then this is an example of CXCL13, which is a chemokines associated with the lymphoid pathotype. As you can see, then you click and you get the distribution, which is obviously in the lymphoid pathotype. You can click on the clinical tab and you get the clinical data associated, or the radiological data in terms of sharp scores or ultrasound score, and then the clinical response. You can also go to the genome tables, and then you can correlate with various parameters, for example, CCP positivity, what are the genes associated with that, or the inflammatory markers. And you can do this both in the blood and in the synovial tissue. So the website link is indicated at the bottom of the slide. So please go and interrogate this database for your preferred gene of interest so you can advance your research and provide, hopefully, better understanding of the disease pathology to the scientific community. So RNA sequence is clearly not a technique which can be used in routine clinical practice. So in collaboration with Genentech, we took the and NanoString, we took the most expressed gene in the various pathotypes and made nanostring panels and reprobe the cohort. And what it emerged, this is another paper we published again in 2019, so I'm not going to spend too much time, but take the headline of this paper, that at baseline, the lymphomyeloid pathotype predicts X-ray progression at 12 months with an accuracy of approximately 92%. So this is a very important discovery, because if we can really see which patients are going to develop a very aggressive disease, then that's where we need to focus our efforts in terms of aggressive therapies for these patients. Vice versa, what's emerging in this patient is that the post-immune pathotype inversely correlated with treatment response. As you can see here, the genes are going down for the lymphoid and myeloid pathotype, or the fibroid, if anything, they are going up. And this makes sense because currently the synthetic DMARDs that we use, including methotrexate in particular, are immunosuppressant medications. But as I showed you at the beginning, these patients, the post-immune patients, they got very little in terms of immune infiltrate. And therefore, why use immunosuppressant medications in these patients? They are unlikely to respond. Most importantly, we also identified a gene set, and this is the third paper that we published last year, that at baseline can predict which patients are going to do badly. In other words, they're not going to respond to synthetic DMARDs, and they end up at 12 months on biologic therapies. And when we look at a small cohort of these patients that were followed up longitudinally, and they went on to anti-TNF therapy, you can see that, again, the fibroid pathotype here at the bottom, 70% of patients who go on to anti-TNF therapy, they do not respond to these biologics. So we have identified now a number of cellular and molecular pathways which are going to tell us that it's a waste of time and waste of money to give anti-TNF therapies to these patients. And there is a huge unmet need for the development of new drugs which need to be targeted to these patients. So the stromal cell reaction is now, through the works of Mark Mike Brenner and Chris Buckley in particular, the stromal reaction are becoming critically important in driving inflammation, and therefore this is where the pharmaceutical industry should focus their effort to develop drugs for these patients. Next program, I'm not going to tell you very much because we locked the database last week. But what I like to say that this was a real European effort, and I'm very proud that we, together with many wonderful investigators, 27 sites across Europe, with the support of the MRC, but also with interest from pharmaceutical industry in the research that this program will lead, we deliver a full randomized clinical trial, biopsy-driven at multi-center level. And we over-recruited to the trial. And now we have 226 patients in this trial, which are going to be analyzed very soon. Let me tell you the paper that has been just accepted in the Lancet. Still, this data is embargoed, so this is not for additional dissemination. But I like to really show you what, in fact, we presented at the ESCR as well. And this is the first randomized biopsy-driven clinical trial in patients inadequate responders to anti-TNF therapy, which were randomized to rituximab or tocilizumab. And the hypothesis that we were testing here is that, as I mentioned earlier, approximately 40% of patients do not have any B cells in the joint, and therefore 60% of patients do not have any B cells in the joint, only 40% have. Why give rituximab to this patient? And the primary trial hypothesis was that tocilizumab, our active control, would be superior to rituximab in these patients. While the secondary hypothesis, for which we did not have sufficient power, and the trial was not powered for this, is that in B-cell rich patients, rituximab is non-inferior to the tocilizumab. As mentioned, the trial was powered only for superiority of rituximab in the B-cell poor population, according to an intention to treat analysis. The primary endpoint was an improvement of 50% in clinical disease activity index at baseline or from baseline. But patients were also considered non-responders if they don't reach a CDAI improvement of 50% and the target CDAI of less than 10.1, i.e. they were in low disease activity, and we define this as major treatment response. These are the secondary outcome measures, which I will not go into detail. And as mentioned, the study was not powered to test the secondary hypothesis. So this is the trial design. Patients were screened and then biopsy and randomized to either rituximab or tocilizumab, 1-to-1 randomization. The primary endpoint was assessed at 16 weeks, and then the responders were continued on the same drug, whereas the non-responders were switched to the alternative drug. So it was crossover design. The classification of patients in B-cell rich and B-cell poor was in a sense double. It was defined as the histopathological classification or the molecular classification here on the right. You can see that when we look at the histopathological classification, although the CDAI 50% did not reach statistical significance, but we do have a significant response when we look at the CDAI-MTR. Twice as many patients reach CDAI-MTR when treated with tocilizumab compared with rituximab in these patients. The results are further emphasized when we look at the molecular classification, which we think is more accurate because instead of looking just at the CD20 expression levels by histology, we are looking at the molecular panel of 73 genes, which are derived from the Phantom 5 gene sets. And as you can see here, both the primary endpoint and the CDAI-MTR were reached with 4 times as many patients responded to tocilizumab compared with rituximab. And only 12% of patients respond to rituximab in the B-cell group, which really is very important to consider that 88% of patients given these drugs were not going to respond. And now if we were to confirm this in other trials, this would be really a major importance for transforming clinical practice. Most of the secondary endpoints were significant, particularly using the molecular classification, which we believe is going to be the future for further trials. There was no difference as predicted, although the study was not powerful in the B-cell rich population between rituximab and tocilizumab. As shown also in this table, for all secondary endpoints, there are virtually no significant difference in any of the parameters that we consider. And now we are busy, this is unpublished data, defining what the molecular profile is for response and non-response. As you can see, we got a really structured heat map here in which we can distinguish the responder from non-responders. We have identified a number of gene sets, and when we put them in prediction model, we can really predict a response to rituximab in particular to the 92% accuracy. So in conclusion, patients with low or absent B-cell linear signatures in the synovial tissue, tocilizumab is more effective than rituximab in this trial, whereas in the B-cell rich patients, they are equivalent. The RNA sequencing analysis reveals specific gene sets associated with response and non-response with good predictive value. And as mentioned, if we were to replicate these studies in other cohorts, this may have profound implications for the management of rheumatoid arthritis in the future. And importantly, there were no serious adverse events related to the synovial biopsies. So the mechanism by which then we can really define response and non-response in these patients could go in different ways. And this trial supports the hypothesis that different disease endotypes may determine treatment response. In other words, if you are B-cell rich, then you give rituximab to this patient, the patient responds. If you are B-cell poor and you still give rituximab, which is called drug A, the patient doesn't respond. Of course, it's not that simple, and non-response can be related to another of other mechanisms, including anti-drug antibodies. So this is, although very unlikely for rituximab, the phenotypic plasticity is another important mechanism, and that underlines probably the secondary non-response. In other words, if you block one pathway, the patient initially responds, but there are escape mechanisms, and we have seen yesterday in cancer, that might lead to a non-response. Very interesting, however, what we have done in the early arthritis, and these are the last couple of slides, really underpin the need for changing the way in which we do clinical trials rather than recruiting all comers. We have identified that patients with early disease can be clustered according to the drug target modules that we are currently using, so B cells, TNF, IL-6, etc. And that leads to then to the next notion that we've learned again from cancer, that these trials, the STRAP and the R4 array, lead into the stratified biomarker discovery element. And we reported this in this review in Nature Reviews of Rheumatology, towards patient-centric precision medicine, similar to what's happening in cancer, where you might have in the same type of cancer, lung cancer, say, one mutation or one pathway, which associated with that patient. And the same will apply for the disease that we want to investigate. So in the umbrella design, we have one disease, different molecular profiles, as I've shown you, and therefore we could really design trials in which we're using the biomarkers to drive drug allocation. We also know, similar to cancer, that, for example, the TNF pathway is active across multiple diseases, including rheumatoid arthritis, psoriatic arthritis, ankylosing spondylitis, so, but not in all patients. So we could define the molecular profiles in different diseases and then use the basket trial design, which will have a major advantage of doing one trial for a specific disease for a specific pathway, rather than having to do multiple trials for one pathway in multiple diseases. For example, the licensing of anti-TNF therapies require specific number of trials for rheumatoid, others for psoriatic arthritis, and so on. So in the future, we might be able to do one trial which will target multiple diseases. And that's what we are going to do in the next venture, which is funded by the European Communion. This is the IMI program, the 3TR program, Taxonomy, Treatment, Targets, and Remission, which started approximately 1 year ago. And we are using this platform to design the next trial in which we are using the pathway-driven approach to allocate therapy. And I hope the future is bright. And I'd like to thank all the people who have contributed, all the collaborators, particularly the investigators in our 4RA and STRAP, and my team, and above all the patients who agreed to participate to these trials. I'd like finally to thank you for your attention and happy to take any questions.
Thank you very much, Kos, for this comprehensive overview and for this outstanding insight in precision medicine in rheumatoid arthritis. I particularly like the concept of the tocilizumab versus rituximab, which I think is the first time really you show that tissue differences are related to drug responses, which I think is very nice. There's one question from the audience. Did you perform synovial biopsy in patient receiving JAK inhibitors?
We have a trial ongoing, which we call phospho-TOF array, and we are taking biopsies from these patients prior to giving them tofacitinib. I can, again, preliminarily share, not from that trial, but from the early arthritis cohort that we tested for the phosphokinome. And again, we have, not surprisingly, a diversity in these patients too, which again will indicate that the underlying kinase activation and expression levels in these patients may determine treatment response, which again clinically explains why not all patients respond to JAK inhibitors either. So we're going in the same direction.
So thank you very much, Kos. I think we have to move on, and I will hand over to Gerhard Krönke, who is my co-chairman. He will introduce the next speaker.
Yeah, good morning. So I have the pleasure to introduce John Isaacs from Newcastle University. John Isaacs contributed very important work to precision medicine in rheumatology. So he identified several biomarkers, and that's one focus of his research in different rheumatic diseases, and recently also developed very innovative approaches for actually tolerance-inducing therapies, including dendritic cells as potential approach to induce tolerance in autoimmunity and rheumatoid arthritis. So we are very much looking forward to his talk.
Thank you very much, Gerhard. Can you hear me okay?
Yes.
Yes.
Yeah, great. Thank you. Well, look, thanks very much for the invitation. I'm presenting this talk from my kitchen this morning, as you can maybe able to see. And this, I was delighted to be invited to this symposium, except that once I'd accepted the invitation, I realized I was going to be speaking directly after Costantino. So it kind of, I thought there was probably little point in talking about some of our own work after what I knew was going to be such a fabulous talk on synovial biomarkers. So I've taken a slightly different approach, and what's going to follow is a bit of a literature review, and it focused on what I'm calling the precision gap, and you'll see what I mean by that. So, you know, I think to a certain extent, precision medicine and oncology started 20 years ago, really, with HER2 and targeting HER2 in breast cancer. We're 20 years down the line, and you just heard from Kos where we are with synovial biopsies. So we're quite a way behind, I would say, in terms of being able to provide the best treatments to the appropriate patients. And I'm calling that the precision gap. And what I want to focus on in this presentation is a few ideas as to why that precision gap exists. And I've got them all listed on this slide. And I'm going to start off with pathogenesis. And it's important to know your pathology. And the reason I'm focusing on pathogenesis first, this is probably a fairly simplistic view. But, you know, in cancer, we know to a certain extent where the problem is. It's in the malignant cell. And so if you can find problems with that cell and target them, and those problems vary in different disease types of disease, then that's a good start for precision medicine. The rheumatology world, like many other disease areas, has been transformed over the last 2 years, largely with single cell technologies. And this is one of the first papers that came out of the so-called AMP consortium, which is mainly based in America, but also in Europe. And this is a single cell experiment on rheumatoid synovium. And you've just seen Costantino's slides, which are lovely, beautiful slides showing the 3 main pathotypes in rheumatoid. But what this paper shows, for example, you know, we heard about Palsy immune, but there's at least 4 types of fibroblasts in the RA synovium. There's at least 4 types of monocytes. There's a large number of T cells and B cells. And we were probably most familiar with these before this work came out because we're largely immunologists in the rheumatology sphere. And here we've got individual patients, both what's called leukocyte rich and leukocyte poor, but you can see each patient has a very different proportion of the fibroblast subsets, the monocyte subsets, the T cell subsets, the B cell subsets. And the problem is we don't really know which of these cells are driving the disease. And so unlike cancer, we're still, to a certain extent, searching for the disease target, if you like. This is another paper from last year from Adam Croft in Birmingham working with Chris Buckley. And this is focusing specifically on the fibroblast in rheumatoid. And they demonstrated that there were 2 main types of fibroblast, at least in this paper, of importance. And they showed that there are so-called lining layer fibroblasts, which are Thy1 negative, they're in blue here, and Thy1 positive fibroblasts. Actually, the Thy1 are in the sublining layer and the Thy1 negative are in the lining layer. And what they showed is that Thy1 negative, the lining layer fibroblasts, contribute very much to bone destruction in rheumatoid, whereas the Thy1 positive contribute very much to inflammation. So when they induced arthritis and then intra-articularly injected either Thy1 negative or Thy1 positive cells, what you can see, the Thy1 negative cells led to more bone destruction, whereas the Thy1 positive cells led to more inflammation. So there's, just for a start, a cell that we really thought was pretty unimportant in rheumatoid arthritis has a very clear key role in determining inflammation versus destruction. This is another paper on the fibroblast, an even more recent paper from this year, showing the increasingly sophisticated nature of the disease that single cell sequencing is revealing. So this is from Michael Brenner's group in Boston. And what we're showing here is spatial transcriptomics, showing that there's a gradient, if you like, of transcription from the sublining fibroblast to the lining fibroblast. Again, these are the Thy1 positive, these are the Thy1 negative lubricin, and this is lubricin expressing. And there really is a concentration gradient in terms of transcription profile from the sublining to the lining. And in this paper, what they show very neatly is that there are signals coming from the endothelial cells here in pink, particularly the arteriolar endothelial cells, which seem to program the fibroblast. And the further the endothelial cells, or the further the fibroblasts are from the endothelial cells, the less likely they are to express Thy1. And that, it seems to be this is responsible for that gradient of expression. And indeed, it seems to be a Notch3 signaling pathway. So their fibroblasts express Notch3, and they interact with Notch3 ligands on the endothelial cells, again here in red, and that leads to Thy1 expression, for example. Interestingly, it also leads to Notch and Notch ligand expressions in the fibroblast, so it leads to this gradient. So I just show this as an example of the increasingly sophisticated pathology we're recognizing in rheumatoid. You've heard about myeloid cells, and this is an even more recent paper just from a couple of months ago from colleagues in Glasgow, showing a number of myeloid subsets in the rheumatoid synovium. Again, single cell sequencing. And the key here is at the top here on the right, showing that patients with RA in remission have a lot of these myeloid cells or macrophages, which are MER-TK positive, CD206 positive, and patients with resistant or refractory disease have much higher concentration of double negative cells. And furthermore, if you stop the drugs in these patients in remission, the more of the double positive cells patients have, the more likely they are to stay in remission. So these double positive macrophages, MER-TK positive, CD206 positive, seem to have a real prognostic value in determining the course of rheumatoid arthritis. And here, histologically, you can see these double positive cells in remission biopsies in the lining layer. They're still present in inflammatory tissue, but they're much more scattered. And in some inflammatory patients, they're completely absent. So that again, it's not black and white. There's different proportions of these double positive cells, but in remission, they tend to be very much in the lining layer and they dominate. Furthermore, if you do co-culture experiments where you co-culture synovial fibroblasts with these, either the double positive or the double negative myeloid cells, those myeloid cells seem to be able to program the fibroblasts. So if you add the negative myeloid cells, the fibroblasts increase inflammatory mediator expression. If you add the positive, double positive myeloid cells, the fibroblasts reduce their inflammatory mediator expression. So this led to the conclusion that these, and I should say actually, this is simplification. There's actually 9 different myeloid subsets they showed, but they largely cluster into these double negative and double positive. So the double negative myeloid cells produce pro-inflammatory cytokines and alarmins and induce inflammatory response in the fibroblasts, whereas in contrast, the double positive synovial tissue myeloid clusters produce lipid mediators that resolve inflammation and induce a repair phenotype. So yet another example of our increasing understanding. I couldn't let this go by without showing the work of our hosts today in Erlangen. So there's Gerard and Georg. And this is another lovely paper looking at the synovial myeloid cells. And these seem to be a different type of myeloid cell. They seem to be resident cells, so not ones that have come from the circulation, probably myeloid cells that have been seeded during embryogenesis. And another new concept, which I don't know how well you can see this, but these macrophages or myeloid cells seem to form a barrier around the synovial cavity, shown here in red. And if you look at these in the lower right here, there's your barrier. And as the mouse develops arthritis, that barrier breaks down somewhat. And it almost looks as though those macrophage-like cells are extending their pseudopodia into the synovial fluid and trying to clear the neutrophil debris. So, a synovial barrier made up of macrophages is something that we've never contemplated before. As you induce arthritis, the density of these cells increases. This is a 3D imaging technique to show the plane of myeloid cells at the barrier. And here's an experiment where diphtheria toxin is used to knock out, so inducibly knock out those barrier cells, leading to more severe arthritis when induced. So another new concept in rheumatoid pathogenesis. Another recent paper, again, just from July this year, the so-called prime cell. This is from Dana Orange in the Rockefeller. And this was a very neat experiment where she developed a technique such that patients could do finger prick tests every week or so, patients with rheumatoid, and taking microliters of blood, which could subsequently be used for RNA-seq whole genome analysis. And what this meant is by taking weekly blood samples, which were useful in the lab, this means they could interrogate flare. So they could look at the blood just before a patient flared, which is something that's been very difficult to do until then. And you can see in this figure here, that you're looking at disease activity and all the blood samples are taken. So if this is the start of the flare here, you can see we've got a lot of blood samples just before, a lot of samples just after. And once you start interrogating those blood samples, you start to see clusters of genes. For example, here's a cluster that comes up after the patient's flare. This is in peripheral blood, remember. And here are a couple of clusters that appear 2 weeks and 1 week before flare. And to cut a long story short, this cluster looks to be a B-cell cluster in peripheral blood, and this cluster looks to be a fibroblast type cluster. Again, very unexpected because we've never, although interestingly, there have been studies in mice before suggesting that fibroblasts circulate around the peripheral blood and can seed joint tissue. Maybe this relates to that, but just before the patient flares, these fibroblasts appear in the blood. Whether they've come from the joint, it's not clear. We don't know what the derivation of these cells are, but there they are. And so, again, I show this as new knowledge in rheumatoid arthritis that we will now hopefully be able to validate and use in our clinical practice. And finally, just this week, yet another nice paper from Georg's group from Erlangen showing another cell type, again, down to single cell sequencing, the so-called regulatory eosinophil. It's been said for a long time that patients with asthma don't get rheumatoid, or at least there's some sort of inverse relationship between asthma and rheumatoid, and this has been known for 20, 30 years. And in this nice paper, again, without going into a lot of detail, the Erlangen group suggests that if you induce asthma in these mice, and ovalbumin-induced asthma, there are innate lymphoid cells in the lungs which release IL-5, and that IL-5 finds its way to the joint where it stimulates eosinophils. But unlike typical pro-inflammatory eosinophils, these eosinophils have a regulatory phenotype, a bit like those regulatory myeloid cells that we just spoke about. And so the eosinophils in the joint and the lung are very different. And when the IL-5 reaches these eosinophils, they develop a regulatory phenotype and switch off the arthritis. So in these mice, you can switch off arthritis by inducing asthma. I thought what was really neat is they managed to find some patients who have both asthma and arthritis. So there are some patients that have both, and this may reflect the fact that asthma comes in different types as well. And when these patients get anti-IL-5 to treat their asthma, their arthritis flares. So perhaps a confirmation of what you're seeing in the mouse. So I'm sorry, that was a really quick run through of a lot of recent biology in rheumatoid arthritis. But to summarize this part of my talk, I just wanted to emphasize that the pathogenesis of the disease, particularly rheumatoid, but also other rheumatic diseases, is becoming increasingly complex. You know, single cell sequencing is revealing things that we had no idea about before. And I would argue that a better understanding should improve our ability to target drugs appropriately and precisely. Now, COS has done some great work on the Synovium, and hopefully that is going to help us in the future. But I would suggest that there's going to still be quite a long way to go to really get to the real precision that we need. So let's move on, and I'm going to talk a little bit now about the disease. And again, my own disease is really rheumatoid, so I am going to spend most of my time in this lecture overall talking about rheumatoid. But know your disease, and I think the precision medicine field in rheumatology is scattered with not very good studies, and that's not a criticism of the researchers, it's a criticism of the disease, really. So again, if we think of cancer, when a patient presents with cancer for the first time, the cancer may have been there for many years, but it's still a relatively virgin disease as far as treatments are concerned. When we do precision medicine studies in rheumatology, or at least until recently, you know, there's a lot of prior therapies. When we're talking of biologics, there's anti-drug antibodies, which have often been ignored as a cause of non-response rather than the disease biology. Our patients have many comorbidities. They're often on other background therapies such as methotrexate and also therapies for their comorbidities. It's the demographics of the disease vary from population to population. And importantly, it's a heterogeneous disease clinically, and I'll come back to that. You know, not all rheumatoid is the same. Patients have different disease activity, different disease duration. There's indirect effects of therapy. So, for example, if you block TNF, we know that IL-6 goes down. And so you might expect if a patient has an IL-6 type phenotype to their disease, anti-TNF might still help. And so that clouds, that confounds interpretation. And some of our diseases like psoriasis, you might see a different effect of treatment in the skin than in the joint, for example. So our diseases are quite complicated compared with, I would argue, cancer, for example. Just in terms of type of disease, I just wanted to show this paper from Newcastle from my colleague Fai Ng, who works on primary Sjögren's syndrome. And he published this paper last year in Lancet Rheumatology. And basically, Sjögren's syndrome, there are no treatments. There's been a lot of trials. It's a disease that desperately needs treatments. But what is clear about Sjögren's is that patients have very different symptoms. You know, not every Sjögren's patient by any means is the same as another. And Fai and colleagues actually used hierarchical clustering and multinomial logistic regression to classify patients simply purely on their symptoms. So he ended up with 4 clusters: low symptom burden, high symptom burden. This is called dryness, dominant, and fatigue. So patients with a lot of dryness and fatigue, and this so-called pain dominant fatigue. So patients with a lot of pain and fatigue. And that seemed to classify the patients quite nicely. And furthermore, if you look at biological factors such as autoantibodies, immunoglobulin levels, and also molecular signatures in peripheral blood, they tally very nicely with these clinical clusters. And you can see here some of the transcriptional profiles clustering. And so this has led to a stratification tool based very much on the symptomatology of patients. And this is retrospective, but in that paper that I've just shown you, Fai looked at a couple of trials, one of hydroxychloroquine, one of rituximab, which had been negative. Basically, they had not succeeded, they'd not given a positive signal, but showed that for hydroxychloroquine, for example, the high symptom burden patient group would have responded. And similarly, in the TRACTIS trial for rituximab, the dryness dominant fatigue type of patient may have responded at the expense of the others. Now, this is retrospective, and obviously we'd need to do this prospectively, but it's just an example of where things may not have to be as sophisticated as the synovial biopsy, for example, that you've just seen, that perhaps symptoms in some diseases may be sufficient to actually provide disease endotypes which are useful. And I know Fai is about to publish another paper on this retrospective analysis in more detail. I think looking at early disease in rheumatology is really important. So I've talked about all the confounders, and increasingly, as you saw with Koz's work, we are trying to focus on drug-negative disease, but these are still patients often in middle life with other comorbidities. So it's still not completely straightforward, but increasingly we are, with our early arthritis clinics, it is possible to find a lot of patients with drug-naive disease. This is just an example from the Glasgow group here looking at epigenetics. So SERA is a Scottish early rheumatoid arthritis inception cohort. They looked at chromosome confirmation signatures on a modest but sizable group of patients, 86 patients with RA, 34 healthy controls. They used a small number of patients to compare epigenetic signatures in RA and health, then used a few more patients to refine and some more still to validate. And actually what they came up with was a chromosomal confirmation signature that predicted the response to methotrexate pretty well. And indeed, they could validate it in a separate set of patients with a nice area under the curve. One of the reasons I'm showing this, though, is that what was of interest is that this 5-marker signature, there's 5 here listed, 3 of them coincide with known EQTLs in rheumatoid arthritis. So an EQTL around the interferon alpha receptor, one around the IL-21 receptor, and one between IL-23 and STAT2. The other 2 did not coincide with EQTLs, but were around genes which we know to be of relevance to rheumatoid. So again, like everything I'm going to show you today, this needs to be validated, but I thought it was quite a nice paper, and I emphasize it partly because it's on drug-naive patients, which I think helps. So to summarize this short part of the talk, know your disease. When designing precision medicine studies, do take care. I think there's there are lots of confounders that we need to control for. And if you don't, do it at your peril. And for example, disease activity at baseline is a really important confounder that often isn't controlled for. Consider different ways of stratifying. It doesn't necessarily require the sophisticated polyomics in certain diseases. And where possible, study drug-naive disease. Okay, I'm going to talk now about outcomes. And actually, for me, this is one of the most important aspects. And I do think it's an area where we are significantly hindered in rheumatology compared to, say, oncology, where again, your outcomes, I would argue, are fairly black and white. It's not necessarily death versus survival, but disease-free relapse. Time to relapse, for example, is also fairly clear-cut, I would argue. And the reason I emphasized here is because if you're going to really do good precision medicine, you need to know that what you're measuring is relevant. And I'm not convinced that's always the case in the diseases that I deal with. So the link between the measured biology and the clinical outcome has to be well defined. Any algorithm, including machine learning, can only work if there's something to learn. Otherwise, you'll get nonsense in, nonsense out. So if you're looking at the wrong biology in the wrong tissue using the wrong clinical outcome or a noisy, unreliable outcome, you won't get the right answer, you'll get the wrong answer. And perhaps that's why less than 5% of our studies have been validated. So it's not just about numbers, and I worry that we are seeing increasingly sophisticated attempts to stratify diseases like rheumatoid. But if you're using the wrong outcomes, then you're not going to get there, basically. And one of the theories that we're working on at the moment is that maybe risk scores are more useful than absolute outcomes as predictors of benefit. So, and I'm going to give you an example of this in the slides that follow. Now, this is a really quite complex paper, and I apologize up front for that, but I think it's pretty important in terms of what I've just said. So this is from a Japanese group. And they're looking for precision medicine markers for rheumatoid, basically. And I'm going to take you through this as best I can. So they started off comparing rheumatoid patients, 45, drug-naive again, with healthy controls. And this is on peripheral blood again. But they compared for transcriptional profiles, serum proteins, using aptamers, and multi-parameter flow cytometry. And they showed a number of differences, as you would expect, in transcripts, serum proteins, and cell counts. I thought the cell counts was quite interesting. We'll come back to that. They then used machine learning to develop models which robustly distinguished RA from health in terms of transcripts, proteins, and cell counts. These were the top 10 genes. I'm not going to go into the specifics. Interestingly, though, the cell counts, rheumatoid patients on the whole had more neutrophils in peripheral blood than healthy controls and fewer NK cells. They were the largest cell differences. And here are the serum proteins, which are interestingly, again, very much focused around complement. What was interesting, I thought, was that the transcriptional differences largely reflected the cell types in blood. Maybe not surprising, but again, we could measure immunophenotyping much more readily than transcriptional profiles. They then looked at response to therapies, looking at methotrexate, infliximab, tocilizumab. So they used EULAR responses as their outcome measure and looked at changes in transcript profile with methotrexate, infliximab, tocilizumab. And what they're looking at is the move towards normality. In other words, do the profiles after treatment look more normal than they did, or more healthy than they did before? And again, here you can see the change in profiles with time. You can see that methotrexate here in blue doesn't seem to do as much as the other drugs. Over here, this is quite important. Actually, the transcripts, if you distinguish responders and non-responders, actually the transcripts aren't very informative. whereas protein changes and cell count changes were much more informative, and that's across the board. Here for methotrexate, perhaps worryingly for those researchers looking at methotrexate, really the whole blood transcripts were not very informative at all, whereas they were for infliximab and tocilizumab. For infliximab, some of the changes actually looked in the direction of more disease rather than less disease. So that's what the red is here. It's away from healthy control. And again, if you look at the cell counts, you can see that each of the treatments reduce the cell counts towards normal, but again, not significant for methotrexate or either for neutrophils or NK cells. And I won't spend a lot of time on this, but again, what they're doing here is looking at the cell count profiles, the serum protein profiles, the whole blood transcript profiles. When they talk about remission, this means after therapy, the cell count or protein or transcript profiles look more like health than they do like rheumatoid. And I think what this is telling us is that even for a drug like tocilizumab, which clinically got a lot of patients in remission, if you look at responders, actually it still didn't necessarily look like a responder in terms of cell count in this patient or protein or transcripts. This patient did. And the other thing, despite EULAR responses, DAS28 remission is okay, but not CDAI and not HAC. And what this is telling us is the heterogeneity in rheumatoid. If you look at the non-responders, non-response is much more brown, so it looks a bit more predictable. But response, no matter what you look at, is very unpredictable. And in fact, if you look at methotrexate, the only blood marker that might be helpful is the cell count. Even in methotrexate responders, the serum protein signature and transcript signature was not helpful at all. If we come down to this correlation plot here, where red is a good correlation, you can see that serum protein correlates reasonably with DAS28 remission, but actually the blood transcripts, the cell counts really correlate very little with clinical parameters of remission. And this was our own experience actually in the RAMAP project. So this is a complicated paper, but the message I'm trying to get across is that what we measure in rheumatoid in particular may not be particularly helpful from a precision medicine approach, particularly in peripheral blood. So just to summarize this part, the link between measured biology and clinical outcome has to be well defined. The wrong biology in the wrong tissue using the wrong outcome will lead to the wrong answer. And, you know, maybe we should be using molecular outcomes, a bit like Cos is saying, but perhaps we should be looking for those myeloid cells that predict remission or fibroblasts, for example. So maybe rather than having a clinical outcome, we actually should be looking for a molecular outcome and looking what pathways get us to those molecular outcomes until we have a better handle on the clinical outcomes in our diseases. So in RA, I'm not sure we're there yet. And very finally and quickly, because my time is short now, I just wanted to look a little bit at methodologies and particularly at machine learning. And just with this one demonstration, I thought this was rather nice. It's systemic sclerosis. And systemic sclerosis can be divided into largely three types, something called fibroproliferative, inflammatory, and normal-like. And this is a paper where they look at skin biopsies, and they've looked at gene expression data for over 11,000 genes across 300 microarrays from 100 unique patients, and use machine learning to dissect out informative transcriptional profiles that correlate with these three subtypes of scleroderma. And they validate it. It's a really nice paper. And they end up, as I say, with three molecular classifiers, one for the fibroproliferative, one for the inflammatory, and one for the normal-like. They then, having defined that in skin, in this most recent paper, which again is very recent, it's only just come out, they look at peripheral blood. And they, again, using a weighted correlation network analysis, they look at genes that change together in peripheral blood in response to treatment and manage to replicate those classifiers using peripheral blood, and then apply those signatures to patients undergoing stem cell transplant for scleroderma. Obviously, this is a very high intensity treatment that you wouldn't necessarily want to apply to patients who aren't going to respond well. So this was a study comparing hematopoietic stem cell transplantation to cyclophosphamide. Again, relatively small, and it's a small number of patients, retrospective analysis. These are the 3 subsets before treatment in the cyclophosphamide and transplantation groups. What you can see, the normal actually doesn't do very well with either treatment. On the y-axis, it's event-free survival. In terms of the inflammatory phenotype or molecular type, they do better with transplantation, but there's not a significant difference from cyclophosphamide, whereas a fibroproliferative pathotype, actually there is a significant difference here. The transplant patients definitely do better. So, this is retrospective, it's small numbers, but you might conclude that if you're fibroproliferative or inflammatory, you're more likely to benefit from transplantation, whereas normal-like, you may as well get cyclophosphamide because you're probably not going to do any better. So I just thought this was a nice example of machine learning leading down a useful path in terms of a disease that's difficult to treat with few treatments available. So to conclude, I would say there's a precision gap between rheumatic disease and oncology. We're not there yet in our diseases. The explanations include pathologically complex rheumatic diseases, our suboptimal outcome measures, and many confounders in experimental design. And I would argue that all of these features have resulted in largely unvalidated stratifiers. I think the future's bright. We now understand pathogenesis better. People are using imaginative experimental design, as you've just seen from COS, novel response classifiers, and the application of artificial intelligence. And these will lead to more complex stratification data, which should reduce the precision gap. But don't forget, you might be able to classify simply on clinical terms, as in Sjögren's syndrome. But most importantly for me is this robust link between the biology being measured and the clinical outcome, without which we're not going to get too far. So thanks for listening. I hope that's been at least a little bit helpful.
Yeah, many thanks for this very informative and nice overview about precision medicine and rheumatology. So currently I don't see questions from the audience. Maybe I can start with a quick question. So actually, from my point of view, what has not been touched so much here are actually the tools we use in rheumatology, which are also kind of part of precision medicine and stratifiers, which are mostly autoantibodies. Yeah? We are, I think, quite well equipped with the possibility in connective tissue diseases, but also in RA, where we stratify diseases and also outcomes by measuring a large panel of autoantibodies. What is your impression? How well do these modern techniques like molecular profiling correlate to these?
Yeah, no, thanks. That's a great question. I think in some diseases they may be more useful than others, and I think we're learning a lot about the diseases from these autoantibodies. So, for example, in rheumatoid arthritis, it looks like particular subtypes of anti-citrinated peptide antibodies, such as antifilaggrin, may predict lung disease, for example. So I think in terms of what they tell us pathobiologically, they're important. In rheumatoid, I would say that at one stage, for example, we thought that autoantibody positive patients would respond better to rituximab, which they do, but it transpires that autoantibodies actually are more of a prognostic marker. So patients who are CCP positive also respond better to anti-TNF, to tocilizumab, to abatacept, probably because autoantibody negative rheumatoid is just a different disease altogether. So I think possibly the autoantibodies in rheumatoid are less useful in determining treatment response because, as I say, they are prognostic markers. If you're autoantibody positive, you tend to respond better to anything. Talking to my colleagues, I think maybe they will be more helpful in diseases like scleroderma, perhaps, myositis. They're not areas I'm particularly familiar with, though, but I think I agree with you. They could be telling us something much more fundamental about the disease and perhaps their likely response to therapies.
Can I make a comment? Okay, thank you, John. That was a great talk, and you're absolutely right to point out that the disease heterogeneity and all the potential pitfalls related to the complexity of the disease. What I think we really need to do, as the oncology have done, is really to contextualize the single cell profiling with the clinical and treatment response subtypes. So better definition of those clinical subtypes, and the fact that the disease heterogeneity is a problem shouldn't stop us from trying. And I slightly disagree on the notion that cancer, they know where the problem is. In cancer, the mutations are even more frequent. The heterogeneity is even more than rheumatoid. They have multiple mutations, both in the primary and the metastatic tissues. And yet they really define new ways of going forward in terms of clinical trial in particular. And that's where I think the rheumatology community should go. It is hard, it is difficult. But if you think about the outcome measures that we have, they are not good, they are not perfect, but certainly we're doing better for our patients that probably the oncologists are doing. But the philosophy is different. So we need to change our philosophy as well and move forward in the precision medicine space with improvement in both the diagnostic and treatment response measures.
Thank you. You know, I obviously I agree, because that's what I think one of the difficulties in our disease is pain, actually, because we don't understand the pathogenesis of pain very well. But it is a fundamental part of the response measures that we use in everyday practice. So if the markers we look at in peripheral blood or indeed synovial tissue don't measure the right biological pathway for pain, then there's always going to be something missing. But, you know, I think as long as we recognize it, that's probably good. You know, but that's as good as we can get at the moment. And as you know, people are looking at more and more objective markers. So Chris Buckley is hypothesizing that maybe it should be a ratio of cell types in the synovium that's our ultimate outcome measure. And I guess that's what you're saying as well, rather than a clinical outcome measure, at least whilst we're trying to define the precision medicine pathways.
Or refine the clinical outcome measure. For example, the two CDAS, the two component DAS really is making an important contribution versus the 4 component DAS. So we really need to refine the outcome measures as well. I agree.
So gentlemen, so thank you for this nice and stimulating discussion. And I particularly liked also John's comment that machine learning can only help if there is something to learn, which is a very important statement. So I want to move on with the next speaker. It's Gabriela Riemekasten. I hope she's hooked on the symposium, and Gabriela is chair of rheumatology at the University of Lübeck. She has a long-standing interest in scleroderma. Hello, Gabriela, and especially she is interested in the autoimmune component of scleroderma and antibodies against G protein receptors. So it's a pleasure to have you at the symposium, Gabriela. I look forward to your presentation.
Yes, thank you very much for being invited. And yesterday you have heard a lot about antibodies as therapeutic tools. And I would like to convince you that antibodies are very, very well biomarkers, as well as, of course, drivers of diseases. So, you know, probably there are some diseases which are already known to, in which antibodies play an important role in the pathogenesis. This is Graves' disease, this is myasthenia gravis, but also in recent time we have discovered completely novel diseases like the anti-NMDA receptor encephalitis and also the membranous glomerulonephritis. And this discovery has really changed life in our patients, because those patients have now the ability to get treatments, and this improves their lives. So that's why I think antibodies are really an emerging field in autoimmunity and in completely other non-autoimmune diseases. So what I would like to convince you in the next few minutes is that antibodies, particularly if they are directed to G protein-coupled receptors, are part of our physiology and pathophysiology as regulators of our immune cell homeostasis. I would like to convince you that anti-GPC antibodies are important biomarkers also in diseases far from autoimmunity. Anti-GPC antibodies are causative drivers, at least for some diseases, and they can probably modulate a lot of diseases. So this is completely emerging field. And anti-GPC antibodies are really valuable tools, novel, so far under-recognized targets for future therapies as well as for precision medicine. So G protein-coupled receptors, I think these are a very important and interesting group of receptors. They are expressed in nearly all immune cells and very selectively in tissues. So they have different binding sites for different ligands, as you can see here for the PAR receptor, the thrombin receptor. You see here the different binding sites of the ligands, and so they can have another signaling. So they have a lot of interaction with other proteins and also other receptors, and the environmental factors affect their expression and activation status. And without these receptors, we are not able to smell or to taste, for example. So if you go into the cell, so you have the exposome, the internal milieu, and for example, cytokines, microbiome, will all determine the expression of G protein-coupled receptors. They contribute to increased or decreased expression of these receptors. They will modulate receptor-protein interactions, the dimerization, for example, homodimerization or heterodimerization, or they will induce conformational changes. So that means everything what we have in our exposome or in our environment will be translated in the GPCR signature. And if we understand this GPCR signature, if we can read the signature, that could help to understand signaling and the function of cells and pathomechanisms. So we work on antibodies against G protein-coupled receptors, and we have measured these antibodies in different rheumatic diseases as a first approach. And you see here, this line represents the normal values of antibodies present in healthy persons. And in some diseases, for example, in systemic lupus erythematosus, you see most antibodies are increased in this disease. But if you go to other diseases, you see, even if they are autoimmune diseases, you see a different and disease-specific pattern. In systemic sclerosis, there are some antibodies increased compared to healthy donors, others are decreased. And this is also present in rheumatoid arthritis, and even more in GPA, vasculitis, granulomatosis with polyangiitis. And you see here the antibodies which are increased in one disease are decreased in another disease. And this means that in normality, we have levels, so between high and low levels. That means we have normal physiological levels of these antibodies. And that's why we were interested to study these antibodies in healthy persons. And what we have done, and that is, this will I show later, but do this physiological antibodies, do they have any relevance? And this is what we have analyzed together with the Gutenberg cohort with Philipp Wild in Mainz. And we have studied together 5,000 individuals in a population study. And interestingly, it was so that anti-CXCR3 antibodies is a chemokine receptor, also a G protein-coupled receptor. This was a very good marker for all-cause mortality. The marker was the same like the high-sensitive CRP value, and Philipp Wild and his group were really astonished about these good marker sensitivity. And also for cardiac death, it was really a very good marker, the anti-CXCR3 antibodies. And on the other hand, there are antibodies against the angiotensin receptor, so also a G protein coupled receptor. These antibodies were good markers for myocardial infarction, stroke, and thrombosis, and as well as hypertension. Those people that have in the fourth quarter higher anti-angiotensin receptor antibodies will suffer more frequently on these events. So that means these antibodies, at least as marker, as risk stratificator, could play an important role. So we have then applied all tests which were available by a company called Celltrend. So, and this company has generated a lot of ELISAs to identify antibodies against G protein coupled receptors, against growth factors, against signaling molecules, as well as against growth factor receptors. And here in these diagrams, you see lines, and the lines represent correlations of the antibodies. So, and you can easily see here, and these are healthy persons, that there are some correlations between different antibodies, and we have compared the levels of the antibodies. And for example, the angiotensin receptor in healthy person has a strong correlation, a significant correlation with endothelin receptor antibodies. And this is what we have analyzed in the past time, that these antibodies are cross-reactive. And summarizing this for all antibodies, this means we have a network of autoantibodies, and these autoantibodies show interactions with a lot of receptors and growth factors and growth factor receptors. And what you see here is in healthy donors, we have differences between female person and male person, but also in old and young persons. And old male persons have the highest range of interactions between the antibodies and a lot of correlations. But most importantly, and this is just another picture, the hierarchical cluster analysis to show the antibody patterns, we found differences among the diseases. And we have found also in Alzheimer's disease patterns of antibodies which were not known so far. And for example, the clusters were among the dopamine receptors, the serotonin receptor, those closely related with the disease. And even also in ovarian cell carcinoma, we found antibody clusters, particularly those directed to growth factors, growth factor receptors. And in systemic sclerosis, because I am a rheumatologist, we also have identified specific clusters, mainly interacting the G protein coupled receptors, the angiotensin receptor, vascular receptors, and also growth factor receptors. And we also now analyze symptom-specific clusters. So we have really identified clusters, for example, which are important in PAH and which are important for lung fibrosis. So going to systemic sclerosis, because this is a very interesting disease, it is also a heterogeneity. So the angiotensin receptor type 1 is really a hub in this antibody network, and the antibodies cross-react or interact with epithelial growth factor receptors and other receptors we already known from the pathogenesis in systemic sclerosis. So, and at least for the angiotensin receptor antibodies, we have measured the levels in the disease, and you can see here, so in systemic sclerosis, we have increased anti-angiotensin receptor antibodies, which are different, significantly different to normal donors, to rheumatoid arthritis patients with primary Sjögren syndrome, dysmorphia, or primary Raynaud's phenomenon. And in systemic sclerosis, we also have increased expression of the receptor, the angiotensin receptor, as well as the endothelin receptor. That is a hint that the antibodies will reflect the GPCR signature. So, and maybe they are a marker also for the environment. So the antibodies are very interesting and important as a predictor of mortality. So you can see here the angiotensin receptor is probably the best predictor. Those with normal values, and we have done before ROC analysis, So nearly everybody survives, but the others will die with a high probability. And also there are markers, predictors for PAH-free survival or survival with PAH. And also this was a collaboration with Yannick Alanor. Also they can predict digital ulcers. That means they are well designed as biomarkers. But the question which really arises, what is the function of these antibodies? And I have shown you a picture. This is the angiotensin receptor, and this is an antibody. And you see here the molecular weight. So, and if they interact, it will certainly modulate the function, and it will certainly modulate the binding of the ligands. So even if maybe the angiotensin receptor clusters with other receptors, so forming large complexes, the antibody from the steric capability can probably interact with the ligands. So what we have then done is that we have immunized mice with the angiotensin 1 receptor. We use membrane extracts from cells overexpressing the receptor to show or to, with a hypothesis that this will conserve the natural structure of the G protein, so with the transmembrane domains. So, and what we have found is that we have induced functional antibodies because these antibodies were able, for example, to induce a higher rate in myocardial cells, but the immunization has also induced interstitial lung infiltration, and you see here germinal-like structures, mainly on T cells, on B cells. In some of the mice, we also were able to induce intestinal germinal centers and also skin infiltration and fibrosis. That means particularly in organs, in interface organs, we have found some pathogenic changes. And of course, this can show you that we can induce some disease by immunization. But to go more into detail, we have generated monoclonal anti-angiotensin receptor antibodies. We've injected these in the skin. And you see here that we were able to induce large infiltrations in the skin compared to the isotype control, and this was significant. But interestingly, if we have done it repeatedly, we also were able to induce interstitial lung disease. So as you see here in the lower pictures. So that means the antibodies are important for the migration of immune cells. They guide the immune cells into the lung because the lung also has a high expression of the angiotensin and the endothelin receptors. This is what I have shown you. These are just a statistic. And interestingly, if you immunize with both receptors, we see here that this will interfere with each other. We have lower infiltration. And also in vitro, if you do migration experiments, we see that the T cells which migrate through an endothelial barrier completely depend on the levels of the angiotensin receptor. So that means the antibodies are very important for migration of immune cells. So if we go back to the antibody levels in different diseases, you see here in the healthy population, there are some people that have high antibodies, but of course they are not sick. And on the other hand, here in systemic sclerosis, we have a lot of patients have low antibodies, but they have the disease. So that means we need not only the antibodies, we probably need further triggers. And in our immunization experiments, we have given the membrane extracts and the antigen together with Freund adjuvant or incomplete Freund adjuvant driving Th1 responses. So that this can induce inflammation. And this is what we have done. We have compared Th1 conditions with Th2 condition. And you see here on the left-hand side, we were able to induce infiltration, mainly on the vessels, around the vessels. But if we use the same immunization protocol in mice under Th2 conditions, So we were able to induce really occlusive vasculopathy. So, and this is very interesting and very important because nobody knows so far why we have this obliteration of the disease present in a lot of diseases, in diabetes, in renal rejection, and so on. So, but to show you that the antibodies play also a role in other diseases, I will show you also some recent data, what we have got together with Carsten Schöpe from the Charité. And we work, all of us now work on corona. And of course, the angiotensin system is very interesting because the ACE enzyme is the receptor for COVID-19. And you see here the strong interaction with this receptor with the angiotensin system. And that's why we have tried to analyze antibodies against this angiotensin system, including also against the receptor for COVID ACE2. And what you see here, and this is a kind of liquid biopsy, that interestingly, the angiotensin 1 receptor antibodies are not different, but the angiotensin 2 receptor antibodies are increased in COVID-19 expression, and they decrease in post-COVID-19 expression, which are quite well. But in those patients which develop myocarditis or dilated cardiomyopathy, so they are increased. And also the same for the ACE2 antibodies. So that means we have probably antibodies that could be a biomarker for, or can probably predict, and this is of course an ongoing research. We try to get money for this research and hopefully we will get it. So, but that shows you that the antibodies are interesting. So, but coming back to the antibody signature, what we know now is that we have a lot of antibodies, and in fact, we have an antibodyome. And or if our antibodies with the angiotensin-targeting angiotensin receptor, maybe also the other antibodies have also a function. And that's why we have stimulated monocytes with the antibodies. And the monocytes are very interesting because these cells traveling through the tissues, they have a very strong interaction with the environment. They are in interface tissues. And we have stimulated here, in this case, monocytic cell lines, the THP1 cell lines. And if you stimulate these cells with SSC IgG, we can really induce guys which are very important in systemic sclerosis. And you see a CCL4, angiotensin, TGF-beta, those factors which are linked to the pathogenesis of the disease. And on the other hand, if you stimulate with IgG from healthy donors, we can induce completely different proteins, so maybe regulatory proteins. And you can probably imagine if you continuously stimulate your cells, endothelial cells, monocytes with your IgG repertoire, maybe this can also cause diseases or can modulate, of course, diseases. And I think this is a new approach to understand. And even if we use individual IgG from our patients, maybe this is a possibility to have an individualized view to pathogenic mechanisms. So, and that is interesting because by using artificial intelligence, we can use those data analysis with the proteome, with the antibody-induced protein, to discover pathways and cytokines which are important here in this case for skin fibrosis. And we were able to, by partial leaf sequence analysis and artificial intelligence, to identify those cytokines which are important for skin fibrosis and those which are less likely important for skin fibrosis. And we can also induce signaling and to see which cytokines can be induced and what can we do against that. And you see here, even healthy persons, the IgG from healthy persons can induce, for example, IL-8 or also CCL18. CCL18 is a profibrotic cytokine. Nobody knows how it is regulated so far, but here we have shown that the antibodies can regulate this cytokine, and even patients with systemic sclerosis show a higher expression rate. And this was completely dependent on the Fab fragment and not on the FC fragment, as shown here by using Fc gamma inhibitors. So, and we can use the antibodies to stimulate the cells and to identify possible factors or possible therapeutic targets which can decrease cytokine expression. And even if we use gene enrichment analysis, we can probably predict some factors, but we can use, of course, in this system also receptor antagonists or pharmaceutical therapies. So, and by this we have discovered that AP1, for example, is a critical regulator with a high potential in systemic sclerosis. And this confirms animal experiments because AP1 is really one of the best models, the FRA2 model for systemic sclerosis. So taking together these antibodies are, I think, very interesting because they could reflect the individual chronic GPCR signature and also the activation status. They can, and this is dependent on the individual chronic environment or exposome. And of course, the antibodies are part of our individual immune system. Everybody has an own HLA type and for an individual disease pathogenesis. And probably these antibodies are ideal candidates for precision medicine. So that's why I hope that I have convinced you a little bit that anti-GPCR antibodies are part of our physiology and pathophysiology as regulator of our immune cell homeostasis. They are important biomarkers in diseases and far from autoimmune diseases. I have shown this in cardiac death and mortality. So anti-GPCR antibodies are causative drivers in some diseases, and probably they also can modulate other diseases, maybe all diseases, what we can imagine. And anti-GPCR antibodies are hopefully, and this is what we have to prove, novel and so far under-recognized targets for future therapies as well as for precision medicine. So I would like to thank all my colleagues contributed to this work. So, and a lot of people do not believe on those kind of antibodies, but I hope we have now more and more data showing that these antibodies are of relevance. And I would like really to thank Frank Petersen and Xinhua Yu from the Lung Center Borstel, and also Harald Heidecke from the Celltran GmbH, because they have given us all these tests. So thank you very much for your attention. I hope I will get some questions, and I hope I have taken your interest. Thank you very much.
Thank you very much, Gabriele. I really enjoyed this lecture, and thank you for diving us into this world of anti-GPCR antibodies, which is, I think, extremely interesting. I have some questions on the screen. How soon in development would you find autoantibodies in healthy individuals versus patients with early scleroderma autoimmunity onset? I mean, when do they start to appear? Is anything known on that?
Yeah, that's of course difficult. We need a lot of, that's why we have approached, for example, also the Gutenberg cohort. But of course, even if you analyze 5,000 people, very few will develop systemic sclerosis. So, but I think this is, of course, what we can do later on. And of course, we do not think that these antibodies are really diagnostic markers. What I would like to have shown you that we have a certain balance here also with other antibodies, and there's no black and white. And that is, I think, the reality in our physiology. And maybe one in a certain region, one antibody could be very important, and in another region with completely other environment, maybe also another antibody is important. So
anything known about early adulthood or adolescence with these antibodies? Did you ever do studies in children or adolescents?
So we have some pregnant. Of course, the antibodies are part of our physiology, but they are probably different in the function. So, but that is, of course, we have the possibility, but we haven't studied so in young persons. So, yeah.
There is also one question regarding the COVID data, which I think which are very intriguing. That, as you showed, that there are I think it was 81 antibodies, if I remember correctly, in COVID-19 patients.
82 and ACE2.
82 and 82, of course, because that's the receptor.
ACE2.
Yeah, and is there anything known about severity of COVID? Are they linked to severity? I mean, do you see a higher antibody response in severe COVID than in mild disease?
Yeah, no, that is ongoing work. So that is why we need large cohorts. And of course, that is, I think, a very interesting the question. But what we have seen so far is that those patients with severe cardiac involvement after COVID have higher levels or have persistent levels of these antibodies. In other persons, we have decreased antibody levels.
There is one more question. There's a large interest in this field. Do you see also disease correlation of particular antibody isotype subclasses? It's more. I think it's a question mostly related to isotypes and isotype subclasses. Is there anything known on that?
So what we have analyzed IgG 1, 2, 3, 4, and we found these antibodies in all of these 4 classes. So what is lacking at so far, and what we haven't just not done, is to analyze the IgM and IgA levels of these antibodies.
Okay. Thank you very much. So, Gabriela, for sharing this nice data, and I hand over to Gerhard again to introduce the 4th speaker.
Bye-bye.
Yeah. Right, so I've now the pleasure to introduce Arndt Kleier, who's a junior group leader here in Erlangen at our department. And Arndt really was very productive during the last years, and he focused mostly on modern imaging modalities, experimental imaging modalities, MRI. I'm not sure if you can hear me. I think there's a problem in the connection. Now it works. Right, so Arndt contributed really very, very interesting work to imaging in rheumatic diseases, mostly arthritis. And I'm very much looking forward what Arndt can present today. Arndt, it's your turn.
Yeah, thank you very much. Thank you very much, Gerhard and Professor Schett, for inviting me to have a talk today. I mean, the bar is set very high for me after these excellent speakers before me and well-known scientists before me. And I think it's quite difficult now to keep up this pace. I was very fascinated about the first talk about the biopsies. I'm a rheumatologist, a clinician, and I see patients and I do clinical research, and I don't do so much basic research. So I was very impressed by the huge amount of biopsies and the input and the information that those biopsies gave us. My focus is, these are my disclosures. My focus today is on preclinical rheumatoid arthritis, or in other words, you can say very early rheumatoid arthritis. And that was the main research focus of the main research interest for me in the last years. Here you can see images of a patient with late-stage rheumatoid arthritis. On the right-hand side, you can see the clinical consequence, and on the left-hand side, you can see the consequence of the information regarding the bone with erosions, bone density, bone mineral density loss, destruction of the joints. Well, this is late-stage RA for sure, and well, my focus is during the past years was to identify patients with very early stage of the disease, and also to characterize those patients and to build up a cohort. Well, the story about the development of rheumatoid arthritis is that in somehow you have a genetic predisposition, here described on the left side with phase A and B. You have a genetic predisposition. Then there are some stress factors, environmental factors, infection, changes in the gut microbiome that might lead to the break of self-tolerance, where you develop autoantibodies. And from the clinical perspective, those patients, they maybe at that phase, they might present at the hospital or at the outpatient clinic with some diffuse musculoskeletal problems. Maybe they are complaining about stiffness of the joints, which is coming and going. Maybe they have some joint pain, or maybe some fatigue, or some unexplained pain. And when you have a look at them, they have no arthritis, but they might have developed autoantibodies such as CCP antibodies. And these are the patients that we were trying to look at very closely during the last years in different aspects. And we tried to build up a cohort since, I would say, 2011, where we have collected those patients. It's called Individuals at Risk Cohort Erlangen. There are some other rheumatologists around the area who are helping us with referring patients when they cannot detect any arthritis, clinical arthritis. And we've built up this prospective cohort now. Currently, there are about 160 patients within this cohort. The main inclusion criteria are the antibody positivity, ACPA positivity, and exclusion, or the end of those patients for this cohort is arthritis. And of course, this is also an exclusion criteria. Now we are trying to collect different forms of data, such as biomaterial data. We heard a lot of before today in the talks about biomaterial data. We are collecting imaging data, and of course, I think this is very important to closely connect those data that we are collecting to clinical symptoms, what the patients are complaining. For example, painful joints, also rheumatoid nodules, or other, for example, family history. This is all the things that we are collecting and storing those in our database. Now, regarding the imaging approaches, I think there are a few questions to answer. For example, diagnostic value of new imaging approaches, but also prognostic values of those imaging approaches. And we're lucky to have a machine where we can have a close look at joints and bone of the peripheral skeleton. It's an HRPQ CT machine with a resolution about 82 micrometers, about the size of a trabecle of the bone. And you can see in this image that we are able to quantify and also to detect very early changes of this bone. In the cohort is a study that we performed a few years ago when we were just building up this cohort, and we could detect significant changes already in patients who are not suffering arthritis yet, but who are of risk to develop an arthritis, who were anti-CCP positive. This is a work about the finger joints, and here we could detect that those patients were already suffering a significant loss of bone, especially the trabecular network, but also some changes in the cortical bone. With the CT, we have the opportunity to have a look with a very high resolution, but we don't get any information about the soft tissue. This is a study, and this was confirmed also by others. This is a study where we could detect that many patients, about 80% of those patients who presented with a clinical suspect of arthralgia and who were anti-CCP positive, had a Tenosynovitis, and not only in one finger, but also it was not only one tendon affected, but also in quite a few patients, a few tendons were affected. And especially those patients were the ones who were very likely to develop a clinical apparent arthritis and who were classified as RA. And others also confirmed these findings, and they also postulated that the stiffness that the patients are that are presenting and complaining is most likely corresponding to this Tenosynovitis. Now, I would like to show you a case that happened in our hospital. And as I just mentioned before, we had pathologic findings regarding the bone density, but also regarding inflammatory changes such as Tenosynovitis and also subclinical synovitis. This is a case of a young patient. He came to our department complaining arthralgia. We detected CCP antibodies, and on the left-hand side, there's an X-ray image about the MCP joints, and the question is if you can see any erosions. The patient came 1 year later, and we performed an imaging again. The patient still had no arthritis, but was still complaining, changing arthralgia. And if you have a closer look using this high-resolution CT imaging, then you can detect at a certain spot the development of erosions. I think that, and for us, that was very interesting because with this case, we learned that relevant changes not only regarding the trabecular bone, here you can see also the trabecular bone, but also the cortical bone, and especially some areas within the joint are of interest, and we have to have a close look because this is where the damage starts for these patients. And if you look at this bone, then you find at a decent spot, you can see that this erosion developed either in the second or in the third joint. And we performed some images and some other experiments because we saw that in this certain spot, we saw that the bone surface looked different compared to other parts of the joint. And when we performed some anatomical approaches, some experiments with human specimens, then we could see that, especially in the structure between the cartilage and the capsule, this part of the bone is kind of not covered, and this is the place where the first where we thought that the first damage of the bone starts. And we called those changes cortical microchannels, and we could see that those comics somehow represent a communication between the bone marrow and the joint space. Here you can see that on the right-hand side, extreme CT images were dissected, and there we could count and also quantify and verify these channels, not only by HRPQ-CT, but we also, because HRPQ-CT allows a resolution of 82 micrometers, but we also could verify these cortical interruptions using micro-CT in vitro imaging, where we had the possibility to go down by even at 10 micrometer. Well, probably one discussion is what are these cortical interruptions? Are these vessels? In the cadaver study, we were not able to detect vessels right inside the joint. We could see here on the bottom, you can see segmented images after injection of contrast enhanced. We can see that the vessels, we could detect the vessels, but not in the space where we have these cortical interruptions. But interestingly, these cortical interruptions and these cortical microchannels, they could serve also as biomarkers and as outcome measures for rheumatoid arthritis or early stage of rheumatoid arthritis. One study that we performed, and you can see results on the left side, is that these cortical microchannels, they happen to be in healthy controls also, but they are increasing in rheumatoid arthritis. and they are also increasing by age. And on the right-hand side, you can see that, and they were associated with erosions. And on the right-hand side, you can see the prognostic value of those cortical channels, because patients in the early stage of the disease, also in a preclinical stage of the disease, who have a broad spectrum of autoantibodies, they also had a higher deterioration of bone in form of cortical microchannels, and this was in the Kaplan-Meier curve also on the right-hand side a factor for developing an arthritis more early. Now, an outlook on the research that we are just performing at the moment. Maybe you have heard that there's also a new way for PET imaging. It's a fibroblast activation protein marker. And currently we are looking at patients in the preclinical stage of RA. Currently we're performing those imaging techniques in patients who complain about arthralgia, and this is an interesting case of a young patient. She came to our hospital because of a sore throat. Quite a few, I would say 2.5 years ago, she was referred by the head and neck department, and they could not detect any reason for her changes in the voice, but they performed lab tests and detected CCP antibodies, and the patient was referred to us. She had no joint complaints at all, maybe for 1.5 years, and then she said, okay, maybe I can feel some arthralgia and it's coming and going. And I performed ultrasound of the joints and I didn't detect anything. Then we performed this FAPI imaging, and interestingly, she had some signals in her right and also the left foot. You can see on the right-hand side the fusion images in the region of the MTP5 and 1, and also in the region of the calcaneus and Achilles tendon. And I would say 1 or 2 months, and at the time point of this this imaging, she had no complaints. And I would say 1.5, 2 months later, she came to see me again, and she couldn't move anymore. Yeah, which was very interesting case. Maybe this is an opportunity to detect the activation of the tissue just before the development of an arthritis and clinical apparent arthritis. Well, the outlook and research again that is currently going on in regard of this as pre-RA or preclinical arthritis cohorts is the effort to homogenize this data in the cohorts. There are quite a few groups around Europe who are focusing on these questions, but all these cohorts, they have different inclusion and exclusion criteria. For example, this is a research question which is currently running within the RTQ consortium. And another focus are, of course, preventional prospective clinical trials currently running with abatacept, and a trial that just finished was with rituximab. The abatacept trials are about to report probably within 2021. The rituximab trial demonstrated that the onset of arthritis was delayed by quite a few months, but I think there's still there's still a huge need for different preventional prospective clinical trials. Also in regard what we just heard this morning, if we had the opportunity to obtain more and more, for example, synovial tissue, this would add quite a huge amount of information, also in planning those trials. Last but not least, I would like to thank especially my group, the colleagues that I work with. Of course, Professor Schett, Gerd Grönke, Ulrike Steffen, who are we are working very closely with the lab. And yes, thank you very much for the invitation, and I'm happy to take questions.
Yeah, many thanks, Arndt, for this great overview about your work, and work is being performed here in Erlangen, mostly focusing on imaging and early arthritis at risk individuals. Yeah, so I don't see current questions. Maybe one question from my side. I mean, we heard these other great talks dealing about stratification of RA, especially this biopsy-driven stratification approaches in B-cell rich, B-cell poor arthritis, and also this more fibroblast rich or driven arthritis forms. So do you think what can imaging contribute to that, or in which respect is imaging able to kind of detect these changes or these subtypes of arthritis, especially the FAPI-PET would be, of course, interesting.
Yeah, very good question. I think this FAPI-PET needs to have some evaluation. I think that the imaging can contribute in two ways. I think it can serve very nicely as an endpoint. And on the other side, I think if this, for example, this FAPI or the other approaches regarding metabolic imaging, if they are more validated, I think then this could serve as a very good, can have a high chance to detect exactly those population patients that we need to see effects of from therapy.
Can I ask a question?
Yes.
Thank you, and that really was a very impressive talk, and very, very important to try to develop non-invasive methods to classify patients early and develop prognostic biomarkers, imaging biomarkers in that way. Can I ask a question specifically with regard to the early detection of Tenosynovitis in patients with rheumatoid arthritis? And the reason for asking this question is because the Leeds group many years ago, in a sense, built a real general hypothesis, which still stands to a certain extent, that patients with psoriatic arthritis had Tenosynovitis, and that was the start of the disease, whereas patients with rheumatoid had mainly intra-articular synovitis. Now, from your studies in Erlangen, it looks to me that actually also rheumatoid arthritis patients have Tenosynovitis early. Is it time to readdress this question of whether the two diseases are different, or simply was the detection early in one versus the other?
Well, thank you very much for the question. I think not only our group or we, but also I think there's quite a few information about Tenosynovitis in rheumatoid arthritis, and also in early stage of RA, and also in preclinical RA. I think the Dutch groups, they have published quite a few papers to that. And I would not make a difference between Tenosynovitis in regard of PSA or RA. I see when I perform ultrasound in the preclinical stage of RA patients, I see quite a few patients who have Tenosynovitis, especially at the extensor carpi ulnaris tendon. I think it's very, very frequently involved.
Thank you. Maybe we should publicize this concept more, because I still think there are people who believe this divergent pathogenesis hypothesis, whereas probably it's a question of simply of detection methods in the right population. Thank you.
Thank you.
Great. Thank you, Arndt. I don't see more questions. And as we are anyway late in time, I will hand over to Georg, maybe for some conclusion.
Sorry. So thank you, Gerhard. Thank you, Arndt, for this a good discussion. And I'm really happy. I think we have seen really three cracks and one young scientist presenting cutting-edge data. I think that's in the spirit of the Leopoldina, because we wanted to bring actually established people together with young researchers. There's also a poster session within this symposium, and I really enjoyed these talks, and I want to especially thank all the speakers for their outstanding presentations. We will have I would say, a 25 minutes break, and then move to the next big topic in immune-mediated inflammatory diseases, which is gastroenterology, and I think we will have an outstanding group of people presenting that, really cutting-edge researchers in the field of inflammatory bowel disease, and we will reconvene then in 25 minutes. So thanks again for everybody participating in this online precision immunotherapy symposium, and enjoy your break.
So welcome now to the last session of the symposium. It's entitled Biomarker and Therapeutic Strategies in Chronic Inflammatory Bowel Diseases. And together with my co-chair, Markus Neurath, it's our pleasure to chair this session. So then I'd like to introduce you the first speaker of this really highlight session within the symposium. And it's really my pleasure and honor to introduce to you Professor Britta Siegmund. Really my honor, because among other departments, she's also head of the Department of Gastroenterology in the Charité University in Berlin, Vice President of the German Research Foundation, member of the Leopoldina, and also President-elect of the European Crohn's and Colitis Organization. So Britta, I think it's also, of course, my pleasure because a dear friend and really a perfect start to this highlight symposium. Britta, please tell us the development of new therapeutic strategies in the field of inflammatory bowel diseases, and thank you for taking the time to be a part of the symposium.
Thanks, Raja, and thanks, Markus, for the invitation to the symposium. And I would have loved to come personally to Erlangen today. So now we have to deal with the virtual version. And I'll be happy to introduce or to share some ideas about the development of new therapeutic strategies. My disclosures. What I would like to do is to guide you a little bit through our current dilemma in IBD. So we make a diagnosis of IBD, and then first of all, we don't really know what is going to happen over the next decade with the individual patient. And I'm very happy that I do not have to cover this topic because James Lee will take care of this topic in the next talk, and there's no better speaker probably around to address this topic. So James will have to address this. What I would like to do then is to outline what we are currently doing, and it's actually quite frustrating what we are currently doing because this has not really a lot to do with precision medicine. But then I'd like to switch gears and focus on how can we predict response to a selected therapeutic strategy, and this points a little bit into the direction how precision medicine can develop in IBD. And in the last part, I'd like to line out how we currently monitor but how we should monitor probably disease in the future. So let's start with the current strategies for therapy selection. And so we have, and I'm only focusing on the drugs that have been introduced over the last 2 decades. So those are for IBD, TNF antibodies, integrin antibodies, IL-12, IL-23 antibodies, and JAK inhibitors. However, it's only one JAK inhibitor at this point. And the decision we make when we are in the clinic, we make the decision based on disease activity, side effects, and extra-intestinal manifestation. I'm going to line this out very shortly in the next few slides. So when we first consider what is the challenge in the clinic, so we either have a patient with a very high disease activity and a lot of personal burden, then we need a rapid improvement for this individual patient. However, there are also patients that have only mild to moderate disease activity, and here the time pressure is a little bit less. And so depending on this and considering the other factors I have introduced, we make the decision whether we choose anti-TNF vedolizumab, ustekinumab, or tofacitinib. So when we start with anti-TNF, and this is clear to everybody, and shown are the data from the Classic 1 trial for Crohn's disease, we have a rapid response, and those are week 4 data showing clinical remission at 36%, but response really in up to 60% of the patients. So rapid response in Crohn's disease, and this equally reflects for ulcerative colitis, and shown as the Act 1 trial and the week 8 data, and 70% of the patients, as shown on the left-hand side, show a clinical response here. So anti-TNF very fast. How about ustekinumab, so anti-IL-12, anti-IL-23, and shown are the data for Crohn's disease and Unity trial, and Unity 2 being the population that is more naive, has not shown a failure to biological treatment. And here again, we see already in week 6 that almost 60% of the patients do respond. So it may not be as fast as an anti-TNF antibody, but it's still pretty fast. Very similar, looking at the week 8 data for ulcerative colitis, the Uni-Klinikum study, we also see a clinical response in 60% of our patients. So how about the JAK inhibitors? And shown here is tofacitinib, the only currently approved JAK inhibitor for ulcerative colitis. It's not working in Crohn's disease, and it's a JAK1/3, mostly JAK1/3 inhibitor. And shown here are the week 8 data, and you can appreciate, and this is now clinical remission, so a much more challenging endpoint, that close to 20% of the patients are responding. There is a publication by the Amsterdam group indicating that you can appreciate the response within the first 2 weeks. So it also seems to be a rather fast responding, a drug which is inducing a fast response. Vedolizumab, and Sebastian Zundler is going to line this out in much more detail later in his talk, due to the mechanism of action. So by inhibiting the infiltration of inflammatory cells to the site of inflammation, the time to response is significantly longer. And this is shown here first for Crohn's disease, and those are week 6 data. So 30% of the patients did respond. And you can see that there was no significant difference with regard to response in week 6. However, there was a significant difference with regard to clinical remission, and this might be a little bit discussable. However, when we look at ulcerative colitis, there was a response in almost 50% of the patients in week 6. So yes, there is a response, but also from clinical experience, we know that it takes a little bit longer. So when we have to place them, going back to one of the first slides I've shown to you, I think there's no doubt that anti-TNF is the fastest drug we have, and that we're all using for severe cases and for severe inflammation. And then ustekinumab and tofacitinib for ulcerative colitis are on the same level, and as ustekinumab for Crohn's disease. And vedolizumab we all take if we have a little bit more time, and we probably take it a little bit more often in ulcerative colitis. And I'm sure Sebastian will now line out why this makes more sense in colonic disease. So when we look to side effects, and I'll be very short about this, for anti-TNF, we all know that we have more infectious complications, in particular in the elderly. We have data, I'm not going to show them, for vedolizumab and ustekinumab, and this is really low risk with regard to safety. And we are all aware of the EMA warning for tofacitinib. And I like to add a little bit to this because there has been a very nice recent meta-analysis that included all currently available clinical studies on tofacitinib. So not only the IBD studies, but also the studies from dermatology and rheumatology. And shown on the upper part of the slides are the studies for IBD. And summarized below is always the total number of all studies that have been included in this meta-analysis. And I'd like to show first the data for herpes zoster, and we have an increased risk ratio in the IBD population, which is 2.37, and it has been shown that there is a clear dose response effect here, so higher doses leading to more herpes zoster, and that explains why in the overall meta-analysis this risk is lower. So this can be a problem in the IBD population. And cardiovascular events, probably very similar. We have an increased risk ratio of 1.8 in the IBD population, which probably also relates to the higher dosage we have to use for inducing remission, which is 10 milligram BID. So coming to the last point we consider, and I think this is a point all of us very often don't consider, because we are gastroenterologists and others in the room are rheumatologists or dermatologists. We all prefer to focus on the organ we like and we feel comfortable with, but it makes totally sense to ask the patient really about the extraintestinal manifestations, because the drug we might choose for IBD might not solve the problem for the extraintestinal inflammation. And this table lines out a little, or provides a little bit of guidance how we can act, or how we currently act in clinical practice. And the key point I'd like to emphasize here is for Crohn's disease, when we choose ustekinumab, we really have to emphasize and to be aware of that ustekinumab is not working in axial involvement of an SPA in this patient. And I think that's something very, very important. So this is not really at all precision medicine. This is more practical approach to data we currently have. So how could this become a little bit more precision medicine? And I'd like to show a few data, how this might change over the next years. And I'd like to start with a study that has been published by Raja Adria a couple of years back, where they labeled adalimumab, so TNF antibody, and sprayed the anti-TNF antibody adalimumab on the inflamed mucosa, and could then endoscopically detect TNF, the adalimumab binding to TNF in the mucosa. In patients who expressed high amounts of TNF and bound the TNF, the labeled TNF antibody, showed a better response to anti-TNF therapy. Picking up a little bit of this concept, we developed a study in Berlin based on an observation that was actually made 30 years ago, that if you take an entire population, you isolate mononuclear cells, you stimulate those mononuclear cells with LPS, you can distinguish a high TNF producing group and a low TNF producing group in a healthy population. And our hypothesis was that TNF high producers are more likely to show a clinical response in week 6. So we set up this study with infliximab exclusively at this point, and assessed clinical activity by using clinical scores and ultrasound, and took the past medical history, took biopsies, unfortunately not in all patients, and isolated PBMCs and stimulated them with LPS, and assessed response in week 8. So I'd like to focus on the Crohn's disease cohort, because here the difference was much more apparent. And first, to show you that we really have this group of high producers, and the line here is showing the median of this population, and a small population of low producers. And if we assess the clinical response in this group, we can see that all high producers responded to infliximab treatment in week 6. And only a minority of the low TNF producers in the Crohn's disease population did not respond. And when you look here at the specificity and sensitivity of the Crohn's disease population, this is really a remarkable result. And we are now addressing in the next study whether this is equally true for adalimumab and other TNF blockers, and will then proceed to a prospective, to a decision-making trial based on this finding. The other biomarker that has been introduced by Ahmed Hegazi and Nathan West, and Ahmed at this time still being with Fiona Powry in Oxford, what they did, they explored oncostatin M for several reasons, and could demonstrate that oncostatin M, as well as the receptor, is highly expressed in Crohn's disease, in tissue of Crohn's disease, as well as ulcerative colitis patients. More importantly is that if you have a high expression of oncostatin M, then the patient is very likely to be, as shown here, to be infliximab refractory. So oncostatin M very well could serve as a biomarker for this population when we have to make decisions. So those two examples, so the oncostatin M and the TNF expression and production, lines out that there are possibilities that we will have to implement in our clinical practice in order to decide which one of the novel drugs we are going to introduce in our patients. And this is also important because the data are very strongly supporting that whatever drug I introduce first shows the best response in our patients. So coming to the fact, so having decided how we treat our patient in the first line, we need to decide how we want to monitor our patient and how to make decisions, how we proceed over time. So how are we currently monitoring and what are currently our goals or therapeutic goals when treating ulcerative colitis or Crohn's disease? And this is, again, very pragmatic medicine. So we assess clinical remission. So we are still basing all the decisions on clinical symptoms. So no blood in the stool, no pain, no diarrhea, and we reevaluate this after 3 months, and we consider endoscopic remission. However, when STRIDE 1 was published, and this was in 2015, CRP and calprotectin were not included as therapeutic aims, and calprotectin, I'm not going to show the data today, but calprotectin now will be included in the second version of those recommendations. And what was interesting at this point, histology was not a therapeutic aim at this point. And I'd like to focus for a few seconds on endoscopy versus histologic remission in ulcerative colitis. And this was very nicely summarized in this meta-analysis recently published in Gastroenterology, where a number of studies were included where an endpoint of endoscopic subscore of 0, so really normal looking colon, or 1 where you have a little bit appearance or reddish appearance or less vascular structure in the colon. So really, if at all, very mild rest of inflammation. So an MAS of 1, which you find in 43% of patients in clinical remission, the annual risk of clinical relapse is 29%. So it's already pretty low. If we go from MAS of 0, so a healthy looking colon, this annual risk further decreases to 14%. So this is already really low, and when one could say, well, this is enough for me, and I don't need further improvement here. However, if we require persistent so really a histologic absence of inflammation, together with an MAS of 0, then we only have an annual risk of relapse of 5%. So this is really a strong guidance help if we need to decide how to proceed in a patient. But doing endoscopy and I mean, every gastroenterologist loves to do endoscopies. However, going for a few seconds on the patient side of the game, obviously the patient is not in big favor of repeating the endoscopy every 3 months. So we need to introduce novel tools in order to assess clinical activity. And recent data from Germany, from the Lüneburg group, really bring ultrasound to and this might sound to the rheumatologist a little bit simple, but in fact, it's only now that we introduce ultrasound to the international community here. And what they've done is they've introduced in a study patients with ulcerative colitis, and they were treated by guidelines to achieve remission. And this was in parallel by ultrasound over time. And what you can easily appreciate that very common markers, so assessment of loss of ostration, bowel wall stratification, and vascularization, that you can really see improvement over 12 weeks. And this was shown now for ulcerative colitis and was equally shown in a sub-study of the Stardust trial for Crohn's disease. And in Crohn's disease, it was remarkable because improvement of ultrasound could still be shown even if in endoscopy we couldn't show more improvement over time. And to compare this to clinical response, so if a bowel wall thickness was normalized then in the sigma, then 90.5% showed a clinical response. And if this goal could be achieved in descending colon, then 96.4% achieved clinical response. So really showing that this is not only some strange marker we want to introduce, but it's really showing clinical efficacy. I think the other thing we have to highlight, and a criticism for ultrasound is always, well, you need a very experienced person to do the ultrasound. Still, the study was a multi-center study. The study had central reading, and I think this shows and reflects that this can be done in basically every center. But having defined how we monitor our patients, what is the problem, and do we is there a need for change or to monitor our patients, maybe not only from an imaging point of view, but also from a more molecular point of view. And why we have this need is shown, is reflected by the study. It's the infliximab approvals trial for Crohn's disease, the ACCENT-1 trial. And you see the clinical patients in clinical remission in week 30 of this trial, and you can appreciate the decline in remission from week 30 to week 54. So even if we have a very good response, we have a loss of response over time. So something must happen here. So, and when we consider this, and we take this thought to a more formal level, we end up with the distinction James will make in the next talk by being able to define a high-risk group and a low-risk group shown in blue and red here. And so once we have decided that we have a group that needs an escalation of medical therapy, the question is, will this cohort always stay on the same line, or do we need to make a decision over time to change our therapy? So is there a change? And this actually, at the end of the day, this leads to the question whether we have a change in disease biology. Is it just due to the fact that we have insufficient disease control, or are there other explanations? And other explanations, in particular for infliximab, are obviously the production of drug-neutralizing antibodies, a phenomenon that we know that is happening quite frequently. But I think we also need to consider that there might be a change in disease biology. And I'd like to point out the example a study also done by Raja's group. When you have a TNF, expressed on a macrophage, and this is signaling to a T cell, the T cell will drive inflammation. If you block TNF, this T cell will go into apoptosis. So now if this, and Raja has shown this very nicely, if now anti-TNF is not working anymore, this might be due to the fact that now the survival signal for this T cell is not coming anymore from TNF, but from IL-23, because the receptor is expressed at a higher level. So here we have a change of biology in the course of disease, and this reflects that we need a molecular monitoring over time, which is not established at this point. With this, I'd like to conclude. So current decision-making in gastroenterology is still very, very basic. So it's based on disease severity, side effects, extraintestinal manifestations. And I've shown you some examples how biomarkers might be able to drive our decision-making in the future. And very, very similar in parallel, the monitoring is based on clinical parameters, imaging, endoscopy, ultrasound, most importantly, and some biomarkers, including calprotectin. But the vision really is to understand the cause of disease on a molecular way. And this then will lead to a scheme like this, where we have in FLAIR, we apply James' strategy and say whether we have a high risk or low risk patient. Then we have a biomarker that will hopefully indicate which drug we use. And then we come to an important point, and this was a little bit shown by the histology and endoscopy data for ulcerative colitis, when we even might be able to exit again. And I think there is still a lot more work to do, and I'd like to thank you for your attention, and I'm looking forward to the questions.
So thank you very much, Britta, for this really excellent overview regarding really every hot topic now in the field of inflammatory bowel diseases. So, and also I encourage the audience to really pose questions to Britta, to use this opportunity via the chat function. So perhaps I may start being the chair of the session. So Britta, although we have now 4 substance classes approved for IBD therapy, we sort of have reached the ceiling regarding therapeutic efficacy. And one could say, okay, we are treating perhaps too late, but even in the CALM trial with patients with really low disease duration, we were not able to achieve mucosal healing over 50%. So I may use this question and ask you, what are we doing wrong, or how can we improve efficacy?
It's a very good question. I think if I would have the answer, yes, we all would be very, very happy. I think it's very difficult. I think what probably is important, I think this was really important about the Stardust trial, that endoscopy in particular for Crohn's disease might not be the final readout, and this is a frustrating sentence being said by a gastroenterologist. But the transmural healing and the transmural progress and response as defined by ultrasound or MRI might be more helpful. But at the same time, what you lined out, and this is one of the key questions we have to address over the next decade, indicates that we are still missing something at the very beginning. And I think we're not able to define this very well currently.
Britta, perhaps I may add an additional question. So following Raja's question, what do you think about combination therapy, and what type of antibodies would you like to potentially combine? Would one combine integrin blockers plus an anti-cytokine agent, or should we combine various anti-cytokine antibodies? What is your opinion?
So I think a very good example would be a patient with Crohn's disease who responds very well who does not respond anymore to TNF, anti-TNF, and has an axial involvement. There, we might treat him with ustekinumab for Crohn's disease, and this might work very well, but we might need an additional TNF blocker for the spine. But this is then not a combination due to the fact that we don't have sufficient efficacy in Crohn's disease. For Crohn's disease, I would favor, so to improve Crohn's disease or ulcerative colitis, I would actually favor combining rather vedolizumab at this point with either TNF or ustekinumab. But I think all of us would feel more comfortable if we would have biomarkers indicating what is actually the best combination. And I think we don't have those markers at this point.
So perhaps one more question from my side, Britta. So we were talking also about endpoints, and you also introduced histology as probably one of the upcoming endpoints. But what do you think in the field of histology, what is still unanswered, like where to take the biopsies, how, I would say, continuous is the inflammation and ulcerative colitis? So what do you think, before we can really implement histology in clinical practice as an endpoint we should reach, what questions do we have to really answer sufficiently?
So I think, first of all, we have to emphasize that this is really an endpoint for ulcerative colitis, but not for Crohn's disease. I think this is important. And where does histology at this point play a role in clinical practice? And I think it plays a role, as I lined out, in exit strategies. So if I have histological healing for whatever reason, as a natural cause of disease or induced by treatment, then I think we can decide to de-escalate or even stop treatment. We lack data actually where we make decisions on histologic healing. So prospective trials, but they will be very difficult to perform. And I wouldn't escalate therapy no matter what, just to achieve histologic healing at this point.
Okay, thank you very much, Britta. So I think, yeah, we have answered all the incoming questions. So thank you again for taking part in the symposium. And of course, yeah, hope to see you again soon, personally also. Thank you, Britta.
Yes.
So then I'd like to introduce now the next speaker to our symposium, and we are heading over to the UK, to Cambridge, and I'm calling Dr. James Lee, and we have already heard about his data in Britta's talk. So James, can you hear me? Excellent. So James, we can see you and I think hopefully also hear you now. Thank you again for taking time, and we have already seen some data of your really seminal work in this field, and now we are eager to hear your insights regarding the prediction of the disease course in inflammatory bowel diseases, which really is one of the most unanswered questions still in clinical practice. So thank you, James, for taking the time to take part.
No, not at all. Thank you very much for the invitation. It's a pleasure to talk to you today. I very nearly wasn't talking to you today because about 40 minutes ago I had a power cut at my house, so I've had to dash down to the lab to try and find a Wi-Fi source. So apologise if I'm a bit sweaty on the way. Okay, so as I say, I've been asked to talk to you today about predicting the course of IBD, which is slightly different to some of the things that Britta was mentioning at the end of her talk in terms of predicting response. So let me start off just by giving you a couple of cases just to illustrate and frame why this is such an important issue. And so these are two real patients, and they both are treated under our care here in Cambridge. When they were both diagnosed, they both had newly diagnosed Crohn's with colonic involvement. Neither smoked, neither had perianal disease, neither had any family history. Both had some mild large joint arthralgias. Both obviously had active inflammation. Probably the guy on the right had a bit more inflammation than the guy on the left. And they were both treated with some prednisolone. So, at this point, these two people are pretty indistinguishable in terms of the way they've presented, at least. So what happened to them after that? Well, let's deal with the guy, the younger guy first, who had a bit more inflammation. So he had his course of prednisolone. Everything got better. He had a follow-up endoscopy a year later where he had mucosal healing. And out to the end of now a follow-up period that's over 5 years, he's not actually had any further flare-ups, and he's not on any treatment at all for his Crohn's. So what about the other guy? So he also had some steroids, and he also got a bit better. But actually, just after finishing his steroids, he had a further flare-up and was started on some azathioprine, and things slowly again got better. But a couple of months later, he then had quite a severe flare-up, was confirmed to have moderately severe disease endoscopically, and was started on anti-TNF. So this is all within 8 months of being diagnosed. But he didn't really respond particularly well to adalimumab, and 3 months later, he was switched over to infliximab. He was actually managed at a time before we had ready access to drug levels and antibody levels. Anyway, he started infliximab, and things slowly got better. But even this is not the end of the story, because a year later he presented with a severe relapse. At this time, we now did have antibodies and levels, and he had good levels with no antibodies, and he was switched over to vedolizumab. But even this isn't the end of the story, because 9 months later, in the context of another severe flare-up, he ended up needing a panproctocolectomy. So you can see the challenge we face. These two guys looked pretty indistinguishable when they first presented, and yet by the time you've gone for a couple of years, you can see how different they are. And this isn't something that's unique to Cambridge patients. This is something that we see all over the world in IBD. And this is a nice study from Scandinavia, where they showed going back to the 1960s, you get some patients who have very aggressive disease, some patients who have very mild disease, and some patients who are somewhere in the middle. And of course, if we're going to do anything to intervene in this sort of situation, what we really want is something that we can do very early on, close to diagnosis, in this sort of window of opportunity, as it's been described, before you get the complications associated with uncontrolled Crohn's disease. And we know that there are potential things we can do. This is from this top-down trial, which is frighteningly 12 years ago now, where they showed that infliximab induction actually improved the outcome in patients compared to sort of conventional step-up treatment. But we also know for several agents now that if you do use more aggressive treatment earlier in the disease course, not only is it better compared to doing sort of standard treatment, but actually it's also better than delaying that aggressive treatment to later on. So you can see this is from a trial of adalimumab, and you can see that your chance of getting a good remission if you have disease for less than a year is pretty much double, that's the 45.8%, compared to if you've had disease for more than 5 years, where it falls to just over 20%. And that's not just a feature of adalimumab. This is a reanalysis of data from the SONIC study, and this is just showing you data from patients who got combo therapy with infliximab and azathioprine, and showing that those patients who had disease for less than 18 months had significantly better outcomes, whether it's clinical remission, normalization of CRP, or mucosal healing, compared to those patients who'd had disease for longer. And it's also not a feature solely of anti-TNF. So here's a study from a vedolizumab trial in Crohn's showing that patients who had shorter disease course actually reached corticosteroid-free clinical remission sooner and more frequently than those who'd had longer disease. So you might then say, well, why not just give everybody early aggressive therapy? You've shown it's better, you've shown it works best if it's given early. Why don't you just give it to everybody? Well, the reason is really encapsulated on this figure, and this is a cost analysis that was published earlier this year, just showing that the costs of pharmacotherapy for IBD patients, particularly biologics, are linearly increasing. And the reality is they're going to continue linearly increasing beyond this as new agents are brought in. Even with biosimilars, these overall costs are going to continue to increase. And of course, we also know that even if most of these drugs are pretty safe, and I think our general feeling is most of them are pretty safe, there are still risks associated with them, and those risks might not be very high. But if you don't need to be taking an anti-TNF, you probably don't want to be taking it either. So this is a situation which, if you went and spoke to anyone in the street in Erlangen or in Cambridge and said, look, here's our situation. We've got some patients who have very aggressive disease. We know that aggressive treatment will work for them, and some patients who've got pretty mild disease. Should we do what we're doing at the moment and treat everybody the same? And they, irrespective of whether they're plumbers or electricians or shop workers, all of them will say, no, you're mad. You can't treat everyone the same. Why don't you just give aggressive treatment to those people who need it and spare the people who don't need it the side effects of those drugs? And that would be an entirely sensible thing to do. And then the question is, well, why aren't we doing it? And the simple answer is we haven't got good biomarkers that can do this. So this, and I'm sure Britta has spoken some about this already, this was the sort of background to which I came to Cambridge 12 years ago, initially to do a PhD with Ken Smith. And the goal of this work at the time wasn't really to find prognostic biomarkers. What we wanted to do was just better understand something about the disease we were treating. And the way we thought we would do this is recruit patients at the time they were diagnosed, before they started any treatment, so you've not got to disentangle the effects of therapy on top. And we would take their white blood cells from different subsets from their blood, and we would profile them using gene expression microarrays. And then the beginnings of this work, this wasn't solely an IBD project. This began in lupus and ANCA-associated vasculitis, and then I followed on with studies in Crohn's and UC. So the first thing that we did when we got this data was we simply said, are there any patterns in the data? So it's essentially like looking at a lineup like this, saying, does everyone look pretty much the same, or are there some people who clearly stand out as being a bit different? And in most of the cell subsets we looked at, it was the former. Everyone looked pretty much the same. And in fact, you can model this by looking at the distribution of genes. So in most of the cell types we looked at, the majority of the genes were normally distributed. The interesting thing was in CD8 T cells, that wasn't true. And actually, there were a large number of genes that had a bimodal distribution. And that means that for these genes, some people are high expressors and some people are low expressors, but there isn't the large population in the middle that you would normally expect to see. And what that meant actually was that when we did a form of consensus clustering, we could identify two clear subgroups of patients. So in this plot, every square is a patient, and it's the same list of patients plotted on the x-axis against them on the y-axis. And all this is showing is that some patients, by their gene expression, always cluster together, and some patients never cluster with others. And those two groups were interesting, particularly because we found that the two patients who I described at the beginning the guy with the mild group disease was in one group, and the guy with the very aggressive disease was in the other group. And in fact, when we looked at this in a bit more detail, we found that actually this clearly associated not just in these two individuals, but across the whole cohort with the clinical course of disease. So we saw an earlier time to needing a treatment escalation if you were in the IBD1 group. That's the sort of high-risk group that's shown in red. And that was true in both Crohn's disease and in ulcerative colitis. And in fact, if we compared how that performed compared to the sort of things we still do in clinical practice, where we look at people and say, well, you're young and you've got perianal disease, and we're needing to give you quite a lot of steroids, so you're high risk. If we compared it with that, or if we compared it with endoscopic features, either in Crohn's or UC, it seemed to have performed better. And it wasn't just time to the first escalation either. So this is a stack density plot shown over time, where you have this is just for Crohn's, the IBD1, the high-risk group are on the right side now, and the IBD2 on the left. And you can see, just as an illustration, that out to 5 years in the low-risk group, about 50% of people have needed no treatment. They're in purple still. Whereas in the high-risk group, that has dropped significantly down. You can see increased use of anti-TNF and immunomodulators. And the same thing was true in UC. So low-risk patients, over 50% out to 5 years, are not really needing any sort of immunomodulator. Whereas, you're starting to see not only immunomodulators and anti-TNF, but you're also starting to see colectomies in the high-risk group. And actually, we've recently shown that in a pediatric IBD cohort, this is also true. So you see, not only can you find the signature, but there is an association with time to first escalation and with number of escalations. So this isn't unique to adult onset IBD. So the question then was, okay, we found this signature and it seems to somehow mysteriously associate with outcome, but why does it? What is actually driving this? And then this was actually some pretty similar work that was done by Owen McKinney in the lab, who showed that if you look at what process is most strongly enriched within the signature, it seemed to be T-cell exhaustion, more so than differences in activation or energy or other things that might be going on. And T-cell exhaustion is something that is actually well described, but not particularly well described in the context of autoimmune disease. So it was originally described as a feature of chronic viral infection, but it can happen with any persistent antigen. And what happens is that your T cells, having originally responded pretty well, actually over time progressively lose their effector functions and begin to express inhibitory receptors. And that means that when they are then re-challenged with the same antigen, they're unable to mount the sort of immune response that you would normally hope they could. And interestingly, if you take a signature of T-cell exhaustion defined in mice and use those genes to cluster either patients with vasculitis, lupus, or IBD, you see that the exhausted cohort, the exhausted group of patients, actually do better in all of these conditions. And if you have less exhaustion, you typically do worse. So one of the interesting things that we noticed around the time was that we were actually getting very similar insights from an entirely separate approach. So this was a genetic study that we'd done, and we were interested in trying to understand whether there might be a genetic contribution to the clinical outcome of IBD. And that's partly because from some very old and nice studies from Oxford back in the mid-90s, it's shown that there are many features of IBD that seem to run in families. So it's not just that the condition runs in families, but often the distribution of the disease and sometimes the behavior of the disease also seems to run in families. And I'm sure if you look after IBD patients, you will have seen this and will have patients you can think of for whom this fits. And so we decided to try and understand whether there might be a genetic contribution to this. And so the way we did this was taking a large cohort of Crohn's patients for whom we had genotype data already, and trying to identify from within that cohort those patients who'd had a very aggressive disease course as defined by the sort of treatments and surgeries they'd required, and compared those with a cohort of patients who'd had a pretty mild course. So they'd had an extended period of follow-up but had never required any sort of immunomodulators, biologics, or surgery. And that was then replicated in a sort of similar but slightly larger cohort. And when we did this, we actually ended up finding 4 genome-wide associations, which is about the right number given the sort of power that we had. And And one of the striking things about this was none of these were susceptibility hits. So these are all very different to the things that are driving the development of Crohn's in the first place. And this is probably best illustrated with the MHC association. So what I'm plotting here, each dot here is a p-value, and I'm plotting the p-values in susceptibility GWAS on the y-axis and on our prognosis GWAS on the x-axis. And the two dotted lines are where you'd, is essentially p of 10 to the minus 5, which is a sort of nominal association that would at least get you interested. Obviously, it's not genome-wide at that stage. But the striking thing is that there are no dots at all in that top right quadrant, indicating that there's not a single SNP in our prognosis GWAS that is also nominally associated with susceptibility and vice versa. And in fact, it was this HLA association that led us to the understanding that there might be something going on with T cells. And in fact, the reason we came to this conclusion was, having identified an association in the HLA, we imputed the HLA alleles and actually found that there were associations with several different HLA alleles. Interestingly, as I say, not the one that's most classically associated with the development of Crohn's. But the really interesting thing was that all of these HLA alleles are known to be inherited together as part of a large multi-gene haplotype. And that haplotype is called ancestral MHC 8.1. And the interesting thing about this, and this is the association of this is with milder Crohn's. So people tended to carry this haplotype if they had milder Crohn's disease. Is that this is already known to be associated with differences in T cell responses. So people who carry this have impaired T cell activation, they don't produce as many cytokines, they have reduced vaccine responses. And if they get diseases like HIV, they have more rapid progression of that disease. So it was all sort of coming, we were getting from two different angles, the transcriptomic data identifying, differences in T cell exhaustion, and the genetic data identifying relative differences in T cell responses, all of which seem to be associating with outcome. And in fact, when we did a pathway analysis, taking all of the hits, and in fact, all of the hits down to a p-value of 10 to the minus 5, just to try and capture anything that was slightly below the threshold of what you'd normally use for genome-wide association studies. We did a pathway analysis and found that the pathways that came out as being significantly enriched for these SNPs were all to do with the regulation of the adaptive and the innate immune responses. And that made a lot of sense, both in terms of, obviously what I've just shown you, but also in terms of our thinking at the time around what is actually responsible for driving outcome in Crohn's disease and other diseases for that matter. So the question then is, can you use the CD8 exhaustion signature to predict disease course? You remember that we need something that can be used in that window of opportunity. And we know that this signature works prospectively. So you take it at the time of diagnosis and it predicts what will happen. So in some respects, we thought this would be a great idea, but then we realized that actually it probably wasn't going to work, particularly if we were going to need individual hospitals to start separating out CD8 T cells and doing microarrays to be able to stratify patients. So we set about trying to do a study to better understand this. And the way we did this was, fortunately, we had, at the time we recruited our CD8 T cell patients, we'd also taken a whole blood RNA tube, which at the time we'd simply thrown in the freezer and thought, maybe that'll come in use one day, maybe not, who knows. And what we did was we tried to find genes in the whole blood that would essentially recapitulate the subgroupings that we'd seen in the CD8 T cells. And so we initially did this with microarray analysis, and then we selected the best genes from some machine learning models that we used in the microarray analysis, validated those by qPCR, and then actually repeated that learning step to identify ultimately a 17-gene qPCR-based classifier that can be done on whole blood. And at that stage, we locked the classifier down and decided to try and find a way to validate this, because there's always a risk in these sort of biomarker discovery studies of overfitting. So your signature looks great when in the training cohort that it's been discovered in, but actually doesn't replicate at all when you then try to look at it in a separate cohort. And so to do this, we aim to recruit patients from 4 different centres, both us in Cambridge, but also St Mark's in London, Royal Devon and Exeter, and in Nottingham. And again, we were after patients for whom they were ideally bled at the time of diagnosis. And this time, instead of having a CD8 T cell sample as well, we just had the whole blood sample. So this was our cohort. We applied our 17-gene qPCR classifier to this cohort, and this quite nicely stratified patients into one of two distinct subgroups, which we termed IBD low and IBD high. IBD low being the subgroup equivalent to the lower risk IBD2 subgroup that we'd seen in CD8s. And then we would have wanted to see what actually happened to those patients over time. And sure enough, we found that the patients who had been predicted to be in the low risk subgroup actually did better. So they had a longer time to requiring a treatment escalation, and that was true in both Crohn's and in UC. And the distribution of treatments they required over time was also different. So again, this is IBD low, so low risk at the top, high risk at the bottom. This is just Crohn's. And again, you can see that out to, this is 2.5 years, patients had about a 50% chance of not needing any extra treatment in the low risk group, but had a much lower chance of managing that in the high risk group. And this was even more striking in ulcerative colitis, where in ulcerative colitis, the low risk patients, nobody needed anything more than an immunomodulator. Whereas in the high risk group, we were seeing not only immunomodulators, but anti-TNF, anti-integrin therapies, and actually colectomies. And it's true to say that across all of the cohorts we've looked at, the only UC patients who have ended up requiring a colectomy are ones who have been identified as being in the high-risk transcriptional subgroup, either in CD8 T cells or in whole blood. So at that time, we actually spun a company out from the University of Cambridge, who are in the process of getting this hopefully NICE approved. And we have also set about doing a biomarker stratified trial. So this is to our knowledge, this is the first biomarker stratified trial that's being done outside of oncology and certainly in any inflammatory disease. And the goal of this trial is to determine whether or not the biomarker would be capable of accurately matching patients to the right treatment. That's either top-down therapy for patients with high-risk disease or sort of accelerated step up for patients with lower risk disease. And if that works, then I think this potentially will end up being a game changer for how we manage Crohn's in the future, because we're not only just going to be able to say, you know, you have Crohn's disease, but you're going to be able to tell people a bit about their Crohn's disease and say, look, we think you've got Crohn's disease, but we don't think you're going to have a very aggressive disease course. We probably can just keep a close eye on you and see how things go. Or we can say, actually, you've got Crohn's and we think it's going to be quite a nasty form of Crohn's, and therefore let's get you onto more potent therapy up front. And although we're using infliximab and azathioprine as part of this trial, there's no reason why it would need to be infliximab azathioprine. It would just need to be whatever is the most appropriate potent treatment at the time. So the trial's underway. It's been going for a couple of years now. We've recruited 273 patients across 42 sites. And hopefully this will be able to finish recruiting early next year. And we should get an answer hopefully towards the end of next year, if not the beginning of the following year. So that's everything I wanted to say. It just leaves me to thank the people who have been involved in this work. I've hopefully called out most of them as they went along. Owen and Daniele have obviously been heavily involved in the work up until throughout the sort of discovery phase. Bram and Nuru, Bram came over from Leuven to spend some time in the lab and was involved in some of the understanding of why we see exhaustion in IBD patients, and haven't had time to go fully into that. And then obviously a lot of this work was done alongside and collaborating with Miles Parks, and it was all done under Ken and Paul Lyons. And so it just leaves me to thank you for your attention, and I'm very happy to take any questions.
So thank you, James, for really giving this excellent overview regarding all the paths that you have taken now to really develop and validate then these biomarkers. And perhaps you can give us again a little insight, because regarding the steps that you now took to develop this biomarker. So first you found it in your center, you went multi-center. So now also looking into the future now, also with the trial that's ongoing. So what do you think that will still need to implement this biomarker really in clinical practice, really everywhere in every clinical center, and perhaps also the advantages of this biomarker regarding applicability and also ease of use probably?
So I mean, I think the reason we did this in whole blood was specifically to make this applicable and ease of use. I mean, there are some very good biomarkers that have been developed, with endoscopic techniques and things, but those are more invasive and patients are less likely to want to have those done on a regular basis. So in terms of the study we've done, we've obviously developed the biomarker, we validated that was all done as part of the gut paper. I think once the question has always been, and the question we always get asked is, okay, you've shown it clearly associates with outcome, but what you haven't shown is whether or not if you had intervened at that early step, would you have actually improved the outcomes for those patients? And that's ultimately what Profile is trying to demonstrate. And I think if Profile does come back and show, a positive result, then, I see no reason why this can't be generally applied for use. Simply because, until there is a better thing out there, it would appear to be the best thing that we have. I mean, we've done quite a lot of comparison testing with other sort of biomarkers, which again, I haven't put into this sort of slide set, but we've not found anything yet that seems to outperform this, particularly at diagnosis.
So James, let me perhaps ask another question. So what does your CD8 T cell signature tell us about the role of CD8 T cells in the pathogenesis of IBD? I mean, most groups have mainly studied CD4 rather than CD8 T cells. They have been somewhat understudied. So what is your opinion on that?
I mean, I think the first thing to say is this doesn't prove that the CD8s are definitely involved. Okay. I mean, it's true to say that if you take a biopsy from patients with Crohn's or UC and you stain for CD4s and CD8s, you will see just as many CD8s as you do CD4s. But you're right that everybody has seemed to focus on CD4s. We also know that some of the best mouse models of IBD are ones that are generated with CD8 T cells that are specific to the intestinal epithelium, and that actually drives a relapsing mouse model, which is quite unusual in some mouse models. So I think directly I suspect that this doesn't necessarily prove that CD8s are the most important cell in IBD. I'm not proposing that they are. We know, of course, that actually the thing that drives CD8 T cell responses in terms of whether people form exhaustion or not, a lot of that is driven by CD4 T cell help. So it may just simply be that what we're seeing in the CD8 T cells is a readout of the other sort of immunological pathways that are involved in IBD and actually involved in T cell responses generally in autoimmunity. It doesn't prove that CD8 T cells are the most important cell in IBD or indeed driving much of this. I suspect it's simply a simple readout of what's going on as part of the sort of immune response to whatever antigen is driving the disease.
So perhaps, James, after showing now all the steps, perhaps a glimpse into the future. So what are the next steps that are planned, also perhaps regarding commercial availability and also now probably awaiting the results of the trial? So what are probably the next steps looking into the future?
I mean, the first thing I will say, which people will find slightly odd, is I'm actually not part of the company. So I do consultancy for the company, but I'm not part of the company. I wasn't a founder of the company. So some of the next steps, I think, are, as I understand it through the company, they have got, and I know some sites in Germany have had some of these tests to actually try on some of their patients to see, whether or not they see similar, associations with outcome as what we've seen. I think the company are clearly keen to try and, make this as broadly available as possible. I think a lot of, certainly regulatory authorities want to see the result of Profile before approving this. And I suspect what we may need to do is actually wait for the result of Profile, and then the company can try to sort of distribute this more broadly. I mean, ultimately, although I am not directly involved in the company, I've seen firsthand that this biomarker works. And it's been great to be involved in its development. So, I would certainly be using it for my patients going forward, obviously subject to being able to use it within the constraints of the healthcare system in the UK.
So again, thank you. Really congratulations again, James, for this really impressive, very impressive work, really changing probably, perhaps in the future, how we treat our patients. So thank you for taking the time, James, and all the best and greetings to Cambridge.
Thank you, you too. Thanks a lot.
So we move on with the program, and before we start with the next presentation, I would like to encourage you to submit further questions online to our speakers. We now move on to the third presentation in this session, and we switch from Cambridge to Oxford, in fact, and it's my pleasure to introduce Fiona Powry to you from Oxford University. She's a leading scientist in the IBD community for many decades and has made seminal contributions to the field in terms of cytokine research, work on regulatory T cells, and more recently on the microbiota. And she will now address the important question, how can we translate our findings from immunology into new therapies for our patients? So Fiona, very warm welcome here from Erlangen. We are very happy that you joined us, and we are looking very much forward to your presentation.
Thank you very much, Marcus, and to the organisers for inviting me to join you today. We're all getting used to this new mode of communication. I've slightly evolved my talk, something a bit more specific, and I want to update you on some of our more recent work dissecting stromal myeloid cell interactions and their role in the pathogenesis of inflammatory bowel disease. Now, inflammatory bowel disease is a hugely complex and multifactorial disease, and that's what's kept us interested and fascinated. And Marcus mentioned I'd been working in this for some decades now, trying to understand the underlying ATO pathogenesis and how we can use that information to enhance our ability to be able to treat this disease. One of the big problems, of course, is it's very heterogeneous. There are the defined Crohn's and ulcerative colitis clinical phenotypes, but even within those, there is great heterogeneity in where the inflammation is, and particularly the pathophysiological features. It's a devastating disease involving young individuals because there really is no cure. As you were hearing from James, many will progress to surgery, and we have a number of treatments, but many will not respond to those. So real unmet clinical need, particularly around treatment refractory individuals. And I want to talk a little bit about some of the pathways that are involved in this. And of course, undoubtedly, this clinical heterogeneity reflects the underlying multifactorial nature of inflammatory bowel disease, which involves a maladaptation between the genetics of the host and environmental triggers that they are exposed to, playing out through the host microbe interface in the intestine. Now, we have made progress, and many in the room or in their rooms listening to this have at this meeting have contributed to this. We have identified collectively as a community a number of different pathways that are involved in the pathogenesis of IBD, notably TNF, more recently interleukin-23, the role of perhaps promoting regulatory T cells, therapies coming online to inhibit the migration and accumulation of leukocytes in the intestine. So we have an increasingly well-stocked toolbox. But of course, there are pathways that we're not covering, and I'll mention some of those. But a problem with that is increasing complex decision-making by clinicians. And what is the information? How can you ascertain which pathway is relevant in which patient? So developing strategies to predict therapeutic outcome will be critical for delivery of individualized patient care. And that relates not just will disease be severe or not, but what pathway is driving disease? So can we more accurately use targets towards particular processes that are relevant in particular patient subgroups? And I want to talk today about some of the later stages in some patients of inflammatory bowel disease with stromal remodeling and intestinal epithelial cell damage. And there really are no therapies that are directed at this area. So our work probing the stromal leukocyte axis really evolved from two wonderfully talented postdoctoral fellows in the lab at the time, Ahmed Hugatzi, who now is working in Berlin with his own lab, and Nathan West, who has established his own lab at Genentech. And they were interested in identifying cytokines that consistently were elevated in both Crohn's disease and ulcerative colitis, and perhaps that had been overlooked in the studies at that time. They came up with four, they trawled the literature in terms of transcriptomic analysis. And of course, this is an area that is increasing, a lot more information emerging for all of us to use in terms of transgene expression profiling in longitudinal type studies. And that's extremely important for hypothesis generation. The risk cohort is one of those that Ahmed and Nathan mentioned, and I just want to call that out as being a fantastic resource for us all that continues to deliver. So what Ahmed and Nathan identified were 4 cytokines, and looking at them, you'll know 3 very well. What took our attention, and certainly Nathan and Ahmed's, was this IL-6 family cytokine, oncostatin M, which we didn't know very much about when they first came up with this. OSM is from the IL-6 family. It binds to the common GP130 IL-6 receptor family cytokine, as well as a unique oncostatin M receptor. OSM is produced by leukocytes and signals into, I'll show you, primarily stromal cells, endothelial cells, and fibroblast populations. But at the time that we started this, there was very little information on its role, particularly in the gut, with evidence that it functions in hematopoiesis, liver repair, and skin inflammation. So as well as showing that OSM and the OSM receptor were increased in inflamed inflammatory bowel disease tissue, Ahmed and Nathan went on to show that high expression of those 2 genes was associated with non-responsiveness to anti-TNF therapy. This is just one of the published studies used, which comes from UC patients that were refractory to steroids or immune suppression and moving on to their first infusion of anti-TNF therapy. Biopsies were taken before that, and I think you can see a highly statistically significant elevation of OSM or its receptor being higher in the patients that were refractory to anti-TNF therapy. And they confirmed this in a number of different cohorts, and others have confirmed this association as new cohorts and information has come upstream. But it's not just oncostatin M and its receptor. Nathan and Ahmed identified a cassette or module, if you will, of cytokines that clustered together that were associated with anti-TNF non-response. And that includes, alongside oncostatin M, IL-1, a number of chemokines. Note that many of these are neutrophil-attracting chemokines, and I'll come back to that, GM-CSF, interleukin-11, amongst others. And high expression of this module was very sensitive and specific in terms of predicting anti-TNF non-response. But of course, is this functionally relevant? Biomarkers are important, and this one would have to be proven in a prospective. But we were more interested in assessing the functional relevance and getting to the bottom of what this meant in the biology. I don't think we're there yet, but I'll tell you about some of our recent studies in this area. In terms of the oncostatin M receptor, Nathan and Ahmed sorted a number of different populations from the intestine and could show that the OSM receptor was primarily expressed on stromal cells, fibroblast populations, as well as endothelial cells. And it elicits quite a complex signaling response in fibroblast cell lines and is quite distinct from TNF, IL-1, or indeed IL-6 itself. Of course, remember OSM is from the IL-6 family, signals through GP130, but the OSM receptor is obviously inducing quite a distinct response, the significance of which we don't fully understand. They went on to show that intervening in the oncostatin M receptor pathway in a mouse model of colitis was able to ameliorate intestinal inflammation. So they treated mice when they had established intestinal inflammation and neutralizing this pathway did not completely inhibit disease. It ameliorated the response and seemed to induce healing in a more rapid way. But more would have to be done to investigate exactly what was going on there. But you can see that this cassette of inflammatory chemokines and cytokines was substantially reduced following the administration of an oncostatin M receptor fusion protein that effectively would mop up oncostatin. So that takes us to where we are now, that introduced a link between leukocytes and fibroblasts, put oncostatin M and its receptor onto the horizon. There was, of course, work from the Greek group on activated, George Kallias, on activated fibroblasts and their role in production of TNF in inflammatory bowel disease. So that's not a completely new concept, but somewhat of an overlooked axis, I would say. There have additionally now been a number of single cell sequencing studies looking at what cells change in the inflamed intestine, and they have identified increases in inflammatory mononuclear phagocytes, some of those producing oncostatin M, confirmed this activated fibroblast phenotype with some specific pathways. And there's also, of course, evidence for activated plasma cells and T cells. But we don't really understand with this mixture the functional interactions and the tissue ecology in IBD in which they operate. And that's what I want to address with our more recent studies, which are unpublished, looking at inflammatory pathways in therapy-resistant IBD, but utilizing a range of different methodologies from whole transcriptomic studies to single-cell sequencing, and quite a bit of histopathology, trying to map gene pathways into tissue ecologies associated with disease. These, of course, give you a snapshot of disease, and ultimately we will want to understand the evolution of any phenotype that we describe. And these now, this study is following on from Nathan and Ahmed's, and this is the work of Matt Jackson, a bioinformatician, Matthias Frederick and Mathilde Pohin, all postdoctoral fellows in the lab. So what they have done is to work with this IBD surgical resection discovery cohort, 100 cases and controls. And this is an example of difficult to treat disease. Matthew performed a weighted gene co-expression analysis, RNA sequencing of that tissue, looked at all of the genes that cluster together. So you get gene co-expression modules, and then looked at how they were co-expressed with different clinical and histopathological features of disease. There's a lot of information in here. I'm not going to go through all of this other than to draw your attention to two modules I'll be referring to, M4 and M5, that were particularly associated with macroscopic inflammation, the Nancy index detecting a particular type of inflammatory architecture. And this was present in both ulcerative colitis and Crohn's disease. And of course, these pathways were at lower amounts in non-IBD tissue. Matthew predicted took these gene expression networks and projected them onto a number of different publicly available cohorts of anti-TNF resistance, as well as corticosteroid non-response and anti-integrin non-response. And for anti-TNF and corticosteroids, the M4 and M5 modules were highly predictive and most associated with non-response to those various therapies. And the same is true for anti-integrin. So this appeared to be a gene expression, a set of gene expression modules that are associated with therapy non-response. So what are the genes that are in those expression modules, M4 and M5? And you won't be able to see this. This is listed, all of the M4 and M5 genes on the y-axis. We sorted distinct leukocyte and stromal cell populations from the inflamed intestine, and then mapped the M4 and M5 genes into particular subsets, into particular cell populations. And a number of the genes associated with these two modules were expressed in stromal cells, and they include, again, these neutrophil attracting chemokines, cytokines from the that signal through the IL-6 family, and a number of other genes to do with stromal cells. And the other portion of genes seem to be almost exclusively expressed in neutrophils. This really gave a very strong neutrophil stromal cell axis. Some of the genes were expressed in mononuclear phagocytes, but at the per cell level, per population level, these markers were most strongly linked into neutrophils, and that includes oncostatin M itself. We think neutrophils are the major source of oncostatin M in the inflamed intestine, alongside a number of other markers that have previously, from the single cell sequencing studies, been linked to mononuclear phagocytes. And of course, what you need to remember with single cell sequencing is that the neutrophil population is lost. So this is just bringing neutrophils into the landscape of therapy-resistant IBD. We went on from there to do some single cell sequencing of the fibroblast compartment, comparing healthy donors and donors with inflammatory bowel disease, isolated the CD45 negative population. These were studies that we did in collaboration with the Roche Fibroblast Network, and Ilya Kurunsky from the Broad played an important role in the bioinformatic analysis. And this has been a very revealing set of data. I don't have time to go into all of it. You can identify a number of different populations of mesenchymal cells, including pericytes, endothelial cells, and two distinct populations of fibroblasts in the healthy intestine, the PDGF receptor alpha positive population, which is lining the epithelium, as well as a population expressing another marker, as well as Thy1, that is deeper into the lamina propria. And others have identified these subsets of fibroblasts in single cell sequencing analysis. Now, in the inflamed intestine, there are a number of changes. And what we've done is, amongst our samples, we have categorized those into, in the inflamed intestine, that have low expression of our modules, M4 and M5, that relate to particular gene expression signature associated with therapy non-response, to those patients that have high levels of expression of M4 and M5. And again, there are a number of changes, but I want to draw your attention to this population of inflammatory fibroblasts, which is present particularly in patients who have high expression of M4 and M5. And you can see that M4 genes in particular map into that inflammatory fibroblast population here shown in red. And if we go and look at the architecture now in the intestine in patients who have high expression of M4 and M5, you see a remodeling of the stromal cell compartment, a particular expansion of these podoplanin high FAP positive inflammatory fibroblasts, which are changing their location and may be playing a role in walling off the intestine as a barrier from the lumen, which can now be exposed because of epithelial cell erosion. You also find these activated fibroblasts. I mentioned to you they're producing high amounts of neutrophil attracting chemokines, and you find them particularly at sites of epithelial erosion and ulceration. Here are the activated podoplanin FAP positive inflammatory fibroblasts in proximity to neutrophil elastase expressing neutrophil cells. So you can see these cells talking to the fibroblasts in a way that one would have predicted from gene and cell expression analysis. What type of histopathology is the M4, M5 high signature associated with? Again, we have categorized all of the patients in that discovery cohort into high, intermediate, or low, and I'll show you a little bit more about that. But in the first instance, the type of histology that is present in the M4, M5 high patients does involve these deep ulcerations with an increase in neutrophils in the M high in red here and a reduction in epithelial cells reflecting epithelial cell ulceration. So this is a population of therapy non-response IBD patients characterized by a gene expression module that we have mapped into a neutrophil and fibroblast activation at sites of ulceration. So is this just a marker of severity, or are there different forms of therapy non-response in IBD that can be identified based on distinct morphology in the tissue, based on distinct histopathology? So we know we can find other modules of gene expression that relate to anti-TNF response, for example, and M6 is one of those, and there are other ones. What type of tissue histopathology and cellular networking do these reflect in the tissue? I think that has been a disconnect from tissue transcriptomics to tissue pathology and biology. So we linked up here with Alistair Easton, a pathologist in the translational gastroenterology unit and oncology centres in Oxford. And he went through all of the histopathology in this cohort of 100 patients and identified and scored them into the different types of pathology that was present: cryptitis, crypt abscesses, granulomas, ulceration, lymphoid aggregates, and goblet cell depletion. And I'm not going to go into all of this, but the M4 and M5 high was associated with ulceration, whereas we identified lymphoid aggregates as being a dominant feature of the M6 gene expression cluster that is also associated with anti-TNF and has a completely distinct gene expression profile. So I want to come back now to this M4, M5 module with the inflammatory fibroblasts and the neutrophil accumulation. We asked the question, what are those activated fibroblasts in the single cell sequencing? What cytokines and various genes are they expressing? And one set of them, as I mentioned before, involves the expression of a number of neutrophil attracting chemokines. So this suggests to us that following epithelial damage, there is an activation of the fibroblast compartment downstream of some sensing and cytokines activating the fibroblasts that then draw these neutrophils into the lesion in an attempt, one would imagine, to try to restore barrier function. So what is the upstream cytokine that is driving that activation of fibroblasts that recruits neutrophils into this tissue eroding lesion? And of course, there are a number of candidates. We used a number of cytokines to look at their ability to stimulate chemokine production, neutrophil attracting chemokine production, the CXCL family in fibroblasts. And the two that were most able to do that were, as has already been described, TNF and interleukin-1. So we wanted to know which is the dominant of those within our tissues from M4, M5 patients that is driving this activation of fibroblasts. So Matthias and Mathilde took resection tissue. They disaggregated that into a single cell mix that led to a conditioned media that they then added to stromal cells, and that gave you very high amounts of these chemokines, these neutrophil attracting chemokines, and that activity was inhibited by neutralizing IL-1 with an IL-1 receptor antagonist and not by adding TNF. So we conclude that IL-1 is a major driver of these activated fibroblasts that then recruit neutrophils at sites of epithelial cell ulceration. And of course, we've wanted to go and map that now into these distinct lesions. And you can see this beautiful localization of IL-1 beta at sites of ulceration that you do not see in other forms of histopathology in the form of these lymphoid aggregates. So I want to finish there. A subset of treatment refractory IBD is associated with an activated neutrophil stromal cell axis. Activated inflammatory fibroblasts produce neutrophil attracting chemokines that we can map to ulcerated tissue. We believe IL-1 is upstream of this cellular interaction, and that maps very specifically to sites of ulceration. Activated neutrophils are a major source of oncostatin M, a previously unrecognized mediator of intestinal inflammation. And this type of crosstalk between the neutrophil fibroblast axis may be very relevant to other inflammatory diseases. So we have identified distinct tissue ecologies associated with therapy non-response in IBD. Clearly, we don't understand all of the upstream events here that lead to the elaboration of interleukin-1. Is that IL-1 alpha or IL-1 beta? But we believe that is acting on fibroblasts to then lead to this remodeling with the neutrophil accumulation. And of course, this does raise the question of IL-1 targeting in a very discrete subset of anti-TNF therapy resistant patients, and of course in more general terms targeting of the neutrophil fibroblast axis for more novel therapeutics. I want to finish there and acknowledge the wonderful collaborators I've worked with over many years, and I've tried to mention those, and of course funders who support this work. Thank you very much.
Thank you very much, Fiona, for your exciting presentation and also for sharing these new unpublished findings with us. Perhaps I may start with a question. So you highlighted the crosstalk between fibroblasts and neutrophils and identified some signals where the fibroblast activates the neutrophils. Is there also a signal coming back? So is this really a crosstalk that further amplifies the inflammation?
Yes, well, the fibroblasts recruit the neutrophils. The neutrophils are making molecules like OSM, of course, Marcus. So it will be very interesting to further dissect that dialogue in terms of what that's doing to the fibroblast population. I think the different interactions that could be involved here have only starting to think about what the IL-11 pathway is doing, a number of these molecules that so far have not been studied as much.
Then I have two questions from the audience. The first question is, do you see an overlap of your IBD fibroblast subpopulation with recently defined cancer-associated fibroblast populations?
Yeah, I'm not exactly sure what molecules that they're referring to there. We're just looking at this across a range of different diseases. That Roche consortium is looking at activated fibroblasts across a range of inflammatory diseases. And there are some shared features as well as some more unique features, and that would be very interesting to look at.
The second question I got is, you demonstrate remodeling of fibroblast populations. What do you think drives this remodeling process? And does remodeling, in your opinion, involve plasticity in fibroblast populations or selective proliferation of individual fibroblast populations?
Yeah, fantastic question. We are getting out of our Bailey here, Marcus, as you would know, the fibroblasts. But it's good to think about some different things. And lineage tracing, we are working now with some mouse models that may allow us to address this question. We did do some trajectory analysis amongst these fibroblast subpopulations, which I didn't have time to go into. And there's no clear precursor product type of trajectory. It may be that activated fibroblasts could come from one or other of these two major fibroblast subsets, but that requires further mechanistic analysis. And I think an area that the field is only starting to consider this type of remodeling.
So, Professor Pauri, if I may also ask one question, I think you have given us really spectacular insights into the molecular mechanisms that drive resistance to anti-TNF therapy on a tissue level, which I think probably is most reflective and most guiding. So do you think these insights could perhaps in the future also lead our therapeutic choices, like to perhaps molecularly stratify these patients and then perhaps also choose the best therapy for these patients, like perhaps even antibodies targeting oncostatin M or IL-1 inhibition?
Very much so. There is an IL-1, related to that, there is an anti-IL-1 therapy going on in acute severe UC. And it will be very interesting to see if we can go back and look in those patients, whether those that did respond had this type of ulcerated phenotype, because it won't be all of them. So not all anti-TNF is the same. I think that's a point. And we've got this large correlative analysis now of different histopathological features across a range of different patients. And if we could employ some machine learning around that to be able to predict then what the cellular drivers are, and I think we don't know that, for example, for the lymphoid aggregate one, but we've got some ideas based on the genes that are expressed. I think it offers opportunity, but it remains to be tested. Prospective studies need to be done, of course.
So Fiona, thank you very much again for your presentation, and we look forward to meeting you in person in the future. Bye.
Thank you. Thank you so much.
So after this presentation, we now move on to the final presentation in this session by Sebastian Zundler from the University of Erlangen-Nuremberg. He is an immunology group leader here at our university and has made some key contribution to the field of immune cell trafficking. And his presentation is entitled Immune Cell Trafficking in IBD, Mechanisms and Therapeutic Implications.
Thank you very much for the introduction, and also thanks to the organizers for inviting me to give this presentation today. Yes, I would like to talk about the mechanisms and therapeutic implications of immune cell trafficking in inflammatory bowel diseases. And what I would like to show you in the next minutes is that this is a field which is really promising with regard to precision medicine, not only in the gut, but there is also quite some way to go to really establish precision medicine here in that field. And maybe also one other remark, in contrast to other mechanisms where we as gastroenterologists rather follow the developments that are set in the field of rheumatology or dermatology, immune cell trafficking is really a field where we are leading due to several new developments which are ongoing. So we have already heard about the multifactorial and complex pathogenesis of inflammatory bowel diseases, so I will go quite quickly over the first part of that slide, illustrating that translocation of antigens from a disbalanced intestinal microbiome over a leaky epithelial barrier leads to aberrant immune cell activation in the gut, finally resulting in under-controlled inflammation. And it's here where most of the drugs available, including conventional immunosuppressants, anti-TNF antibodies, and anti-IL-12, IL-23 antibodies interfere. However, the immune cells somehow have to come to the gut, have to get there, and consistently over the recent years, immune cell trafficking has emerged as a new and orthogonal axis in the pathogenesis of IBD, regulating the abundance and the function of immune cells in the gut. And in particular, the processes of homing, so the extravasation of immune cells from the bloodstream into the tissue, the retention of immune cells within the tissue, and the recirculation of T cells and other immune cells from the tissue back to the blood are the processes that are involved. Well, and that this is translationally relevant is underscored by the fact that several several therapeutic approaches targeting these steps are currently in development or already approved. Yes, first of all, I would like to show you how immune cell trafficking actually looks like in vivo. We have established an in vivo gut homing model in our lab, and as you see here on the left, we inject fluorescently labeled T cells into the ileocolic artery of mice and subsequently analyze the gut homing with intravital confocal microscopy. And I hope that video will also start. Yes, now it works. You see here the position of that green cell marked with white dots. And over the somewhat more than 2 minutes that this clip covers, the cell moves quite some bit into the tissue, indicative of immune cell trafficking in vivo. Now, what are the processes that we summarize as immune cell trafficking? The first step is that T cells and other immune cells may leave the bloodstream and enter into the tissue in so-called gut homing. And here, integrins expressed on the surface of the immune cells interact with cell adhesion molecules expressed on the endothelium. Once in the tissue, several fates are possible. The cells may recirculate either directly to the bloodstream through sphingosine-1 phosphate-mediated chemotaxis, or indirectly through lymph vessels, which is driven by CCR7. Alternatively, those cells may become resident either by CD69-mediated antagonism of sphingosine-1 phosphate receptor signaling or through adhesion of alpha E beta 7 integrin to epithelial E-cadherin. And over the next slide, I would now like to comment on the most important of these mechanisms and the translational aspects that they entail. And you see here some integrins made up of different alpha and beta chains, as well as the respective ligands. So we have vascular cell adhesion molecule 1, which binds alpha-4 beta-1 integrin, and mucosal vascular cell adhesion molecule 1, which binds alpha-4 beta-7 integrin, and those molecules here are expressed on high endothelial venules in the gut. E-cadherin is expressed on the intestinal epithelium and is the ligand for alpha E beta 7 integrin. And the first antibody of this developing family of anti-trafficking agents was natalizumab, a monoclonal anti-alpha-4 integrin antibody, which has first been tested and is still used in multiple sclerosis, but has then soon also been tested in Crohn's disease, where it was found to be effective, but due to severe side effects, due to reduced central nervous immune surveillance, it is not approved for the therapy of Crohn's disease in Europe. However, this was not the end of anti-trafficking agents in IBD, and probably the most important development in this field in recent years was the introduction of vedolizumab, an anti-alpha-4 beta-7 integrin antibody, specifically blocking the interaction of alpha-4 beta-7 integrin with MEDCAM1. And importantly, MEDCAM1 is virtually exclusively expressed on high endothelial granules in the gut. So this is a gut-specific approach with very few or even none systemic side effects. Well, these are also data from our lab where we have been able to show the mechanism of action of vedolizumab in the aforementioned in vivo gut homing model and light sheet fluorescence microscopy. You see here a 3D reconstruction from the terminal ileum. The vessels are labeled in red, and interspersed you will now see several green cells which have extravasated to the tissue. And when we apply vedolizumab in that model, which you see here on the right, then the number of cells arriving in the tissue is substantially reduced in comparison to no treatment, really arguing for a blockade of T-cell homing to the gut following vedolizumab treatment. Well, vedolizumab has not only been effective in seminal phase 3 trials, but has recently also been compared head-to-head, the first ever head-to-head trial of two biologics, IBD, with adalimumab. And here it has demonstrated superiority with regard to the induction of clinical remission and endoscopic improvement, really showing that blocking T-cell trafficking is one of the most effective approaches to target ulcerative colitis. Importantly, this was ulcerative colitis. In Crohn's, it seems to be somewhat different, and actually I didn't have this slide in my presentation, but just added it due to the announcement of Britta Siegmund. So we have been able to show in Crohn's disease, and this is matching the clinical experience, that it might not work as well as in ulcerative colitis, that T cells in Crohn's disease express more alpha-4 beta-1 integrin, and that the homing of those cells in our in vivo model to the terminal ileum is not reduced by vedolizumab, but it is reduced by natalizumab, by pan-alpha-4 blockade, arguing for a model where the blockade of alpha-4 beta-7 integrin in Crohn's disease triggers increased alpha-4 beta-1 expression and ileum homing via another pathway, while this doesn't seem to be the case in ulcerative colitis, where only few cells may enter the colon via alpha-4 beta-1. So despite those very, very convincing results for vedolizumab, and it is now used for more than 6 years very successfully, there have also been some setbacks in that young field of anti-trafficking agents. One example is ontemelimab, an anti-MEDCAM1 antibody, which targets the alpha-4 beta-7 MEDCAM1 interaction on the endothelial side, and although it showed efficacy in a phase 2 trial in ulcerative colitis, the results in Crohn's disease in phase 2 were completely disappointing, and so far we don't really know the answer, but it seems as it is not just a biosimilar of vedolizumab therapy. And then we have the anti-beta-7 integrin antibody etrolizumab currently in development, This blocks both the interaction of alpha-4 beta-7 integrin with MEDCAM1 and also the interaction of alpha-E beta-7 integrin with E-cadherin, thus also epithelial retention of homed mucosal T cells. And somewhat the rationale for trying this has been demonstrated in our lab by showing that the blockade of alpha-E beta-7 and alpha-4 beta-7 integrin led to reduced accumulation of alpha-E expressing T cell subsets in the gut compared with sole alpha-4 beta-7 blockade. And also translational studies investigating the role of tissue-resident memory cells suggested that these cells are drivers of inflammation in inflammatory bowel diseases, since in experimental models, a functional defect of those cells reduced disease severity very strikingly, and in human patients, high abundance of those cells led to early relapse. All of us would have thought that blocking alpha-E beta-7 in addition to alpha-4 beta-7 would be a good thing. However, there have also been other reports showing that the number of CD103 positive T cells in the colon of IBD patients is reduced, and already put some question marks behind that. And very recently, a very ambitious, maybe a little bit over-ambitious phase 3 trial program of etrolizumab in ulcerative colitis has been completed with rather disappointing results. So only 2 out of 3 induction studies met the primary endpoint, and none of the maintenance studies met its primary endpoint. However, the data for some key secondary endpoints were good. So the books are not finally closed over etrolizumab, and in particular, the phase 3 trial in Crohn's disease is still ongoing. Well, then I also mentioned that sphingosine-1-phosphate mediated recirculation via S1P receptors is important in inflammatory bowel diseases, and that's why sphingosine-1-phosphate receptor modulators lead to sequestration of immune cells in secondary lymphoid organs, and several members of this family are currently under development, and particularly ozanimod recently finished a phase 3 trial in ulcerative colitis and showed efficacy both in inducing and maintaining remission. So maybe this might become the next member of the anti-trafficking agent family to be approved. So since we're here talking about precision medicine, I would also like to make some comments on the question whether we can predict response to anti-trafficking agents. And naturally, most of the data relate to vedolizumab. So this is a very interesting work from Ananta Krishnan and colleagues, where they analyzed the intestinal microbiome of patients undergoing vedolizumab therapy. And what they could show was that at baseline, several bacterial strains were specifically up or down regulated in patients entering remission or not. So that suggests that maybe we will be able to predict the success of vedolizumab therapy based on the intestinal microbiota. Those are data from the group of Severin Vermeer in Leuven. They found that 5 genes were significantly differently regulated between patients entering endoscopic remission or not. And when they combined 4 of those genes into a model, they were able to predict remission with an area under the curve of 0.79. And there are also data from Erlangen in that regard. So Rajat Treya's group could show that, similar to his work with adalimumab and anti-TNF antibodies, molecular in vivo imaging by application of fluorescently labeled vedolizumab to the inflamed colon mucosa was correlated with clinical outcomes of the therapy. And then there are also some data with regard to etrolizumab from the phase 2 trials, where the authors could show that both the higher expression of the alpha-E gene at baseline and a higher number of alpha-E positive cells at baseline was associated with significantly better outcomes than low expression. And I already mentioned the rather disappointing phase 3 trial results, but of course it will be very interesting to further analyze the phase 3 data in that regard. So in conclusion, anti-trafficking agents are a very promising new approach for precision treatment of inflammatory bowel diseases. However, we have still several challenges and unmet needs in the field. So we still lack detailed mechanistic insights, for example, how those different therapies differentially affect immune cell subsets. We need to explain those recent clinical trial results to refine our therapeutic approaches, and collectively we already have some approaches towards precision medicine, but none of those has been clinically established, so there need to be further efforts taken, but as I hope I could show you, this is a really promising thing. Yeah, thank you for your attention, and I'm happy to take questions.
So thank you very much for your presentation and showing us the most recent data in the field. So one obvious question is, do you have any good explanation why the etrolizumab data is rather disappointing?
I would like to have one. So I have several ideas. So one thing might be the dosing, maybe. So we know from many drugs from the anti-trafficking agent family that they seem to have a nonlinear exposure-efficacy correlation. So this has been shown for etrolizumab in phase 2, for ontimelimab in phase 2, and also for vedolizumab in phase 2, that very high doses of the drug resulted in worse outcomes than medium doses. And we have some preliminary data on that with regard to vedolizumab, where we think that particular T-cell subsets are more resistant to the drug than others. So maybe this might also be the case for etrolizumab, and in particular with regard to etrolizumab, we have to take into account that things might be different for alpha E beta 7 and for alpha 4 beta 7. So yeah, maybe it's a question of dosing, and the other thing is that alpha E is higher expressed in the ileum compared with the colon, so maybe the Crohn's disease trials will show something completely different.
The second question I have relates to your work on tissue-resident memory T cells. Do you envision any strategy how to specifically target this T cell subset?
Yeah, well, that's ongoing work. So that's something quite obvious that we have to try this. But of course, the prerequisite is that we identify a specific target expressed on tissue-resident memory T cells, but not another T cell subset or other immune cells. So if we would be able to find such a target, then this is a very interesting thing. The problem is somehow that unlike in mice, where TRM cells have a clear, much clearer signature, and also specific transcription factors driving their tissue residency and their development, it is not so clear in humans, and there is a difference to mice. So that's a task for the future.
Okay, I think this concludes our presentation today. So thank you very much for your presentation and showing us the data on T-cell trafficking in IBD. So we are now at the end of our two-day symposium on precision medicine, hosted by the Leopoldina and organized by Matthias Beckmann. And this gives me the opportunity to make some final remarks and discuss with you in short the past 2 days. So we had an initial session on immune checkpoint inhibitors. Laurence Zitvogel told us about the impact of the gut microbiome on tumor immunosurveillance. Dimitrios Myogiakakos on CAR T cells and how we can use them to treat tumors such as leukemia and lymphoma, but also solid organ tumors in the future. Philipp Beckhove then talked about identification of new checkpoints using innovative screening platforms, and Julian Sachs on immune resistance genes and individual co-expression patterns. So this session really made clear that we have many new insights on treatment of tumors via checkpoint inhibitors and CAR T cells, and that we have many options in the future to understand how immunotherapy may advance treatment of patients in the clinic. We had a second session on innovations in tumor therapy. In this session, Barbara Seliger spoke about distinct tumor escape strategies of tumors, lesson for immunotherapy, while Minhard Herlin addressed modeling of human melanoma to understand and overcome resistance against immunotherapy. Finally, Peter Fasching and Anita Kremer from Erlangen spoke about personalized medicine in breast cancer and the analysis of tumor-infiltrating lymphocyte in this disease. So I think these presentations made us clear that we understand more and more about the role of T cells controlling responses against immunotherapy in solid organ tumors. We have many new ideas how to improve precision medicine, and we obtained new insights how to address immune escape strategies of tumors at the molecular level. Today we switched gears and moved to immunotherapy in chronic inflammatory and autoimmune disorders, and we had an initial session this morning about precision therapy of rheumatic diseases. Constantino Pizzalis and John Isaacs addressed precision medicine in rheumatoid arthritis and how we can use modern approaches to guide immunotherapy in this disease. Gabriele Riemekasten spoke about antibodies against G-protein coupled receptors and how we can use them in the clinic to predict response to therapy. And finally, Arndt Kleier from Erlangen spoke about experimental imaging to identify patients suffering from early rheumatoid arthritis that may require early immunotherapy. And then finally, we had the IBD session about biomarkers and therapeutic strategies in patients with inflammatory bowel diseases. Britta Siegmund spoke about new therapeutic strategies in IBD and highlighted new agents that will become available in the future. James Lee spoke about a CD8 T cell signature representing T cell exhaustion that may be used to predict response to therapy. Fiona Powry addressed markers for therapy resistance against anti-TNF therapy and highlighted the role of fibroblasts and neutrophils. And finally, as we just heard, Sebastian Sundler told us about new insights in immune cell trafficking and has highlighted that we have many new agents that will be likely available in the future. So collectively, this data have highlighted that we have many new insights into immunology processes in chronic inflammation and cancer, and that we have many new options to have a more precise medicine, that we can use individualized medicine in the future, most likely by looking into the tissue, understanding immune processes, and understand drivers of resistance against therapy. So clearly, we are now at the beginning of a new era and not at the end, and I hope that you have obtained many new insights into immunology in clinical practice and about precision medicine. And this concludes the current meeting. I very much would like to thank you, also in the name of my co-speaker, Raja Atreya, for your participation and wish you a good afternoon. Bye-bye, and hopefully come back in the future to future medicine meetings on precision medicine from Erlangen. Thank you very much.